Okay, so I guess maybe I'll get started introducing. So we have a minute to say hi. Welcome to the American Naturalist Society Conference session on climate. I just want to really quickly thank the American Naturalist Society. This is my first meeting, and I've just been so incredibly happy and feel incredibly welcome with such kind of like inspiring and principled leadership. I'm so happy to be here. So I want to make one reminder to people who are watching either on Zoom or YouTube. You can actually, is there a question option on YouTube?
We're not monitoring it.
Okay, so if you are on Zoom or in the Slack channel, you can ask questions either directly via our question moderator, Scott Taylor, so via a DM to Scott Taylor, or in the channel for this session on climate. You can also ask questions if you're here in Zoom, directly in Zoom. All right, so it's 12 o'clock here on the West Coast of the US, so let's get started. Our first speaker for today is Matthew Sasaki, and he'll be speaking to us today about plasticity tolerance trade-off across spatial and temporal scales in a widespread copepod. So take it away.
Okay, so hopefully everyone can hear me and see my slides now. So yeah, like they said, my name is Matt Sasaki. Thanks for tuning in, everyone. I'm a new postdoc at the University of Connecticut with Hans Dom. We studied interactions between evolutionary ecology and trophic ecology in copepods. We're just getting that project started now, though. So today I'm going to talk about some of my dissertation work, looking at patterns in the evolution of thermal tolerance and in the evolution of plasticity in thermal tolerance. So like many people, I'm interested in how populations cope with variation in the thermal environment over various scales, specifically in how thermal limits affect or play into this. And so one of the mechanisms that my work focuses on that allows populations to do this through its effect on thermal tolerance is phenotypic plasticity, right? Sort of environmental effects on the phenotype. And plasticity has a really long history in ecophysiological work. And so we know that the strength of plasticity or the slope of these reaction norms can vary both between and within species. And it's these within species patterns that I focus on because they might be really important to understand in a climate change context, right? Since plasticity provides a sort of rapid response mechanism that might help populations endure rapid environmental changes, if the strength of plasticity varies between populations within a species, we might want to incorporate that into the various frameworks we use to assess vulnerability. There's an issue though, which is that there's still uncertainty in regards to what shapes these patterns in the evolution of plasticity, right? So unless we go out and measure plasticity for whatever traits we're interested in all populations of all species, incorporating within species patterns in the evolution of plasticity into predictions about vulnerability is really difficult, if not impossible. But so while there is uncertainty in some of these patterns, again, plasticity has been around in the literature for a long time. And so there are several major hypotheses about what shapes the evolution of plasticity, specifically related to how it affects thermal tolerance or thermal limits. There's two of these hypotheses that I'll touch on today. The first is the climate variability hypothesis, which suggests that plasticity evolves in more variable environments. Plasticity and thermal limits evolves in more variable environments, right? So populations from places with larger magnitude variation in the thermal environment should evolve larger plasticity, a larger capacity to modify thermal limits through plasticity, while populations from more stable environments would have reduced plasticity. The other hypothesis that I'm going to touch on is the trade-off hypothesis. So rather than being sort of directly shaped by patterns in the thermal environment, the trade-off hypothesis suggests that patterns in the evolution of plasticity are more strongly related to, or maybe more strongly constrained by patterns in the evolution of thermal tolerance, right? Where populations that evolve increased thermal tolerance reduce their ability to modify thermal limits through plasticity. And so this is one of the questions that my dissertation work touches on, sort of looking at what shapes patterns in the evolution of plasticity within species, not only over these sort of spatial scales, they're more common, but as well as over temporal scales. So a little bit of background really quickly. I work with copepods. These are the most abundant animals in the ocean, if not on the planet. And so because of this abundance, they're incredibly important for sort of ecology, as well as biogeochemical cycles, especially in the ocean. The species that I've done most of my work with for my dissertation is this one, Acacia. It's widespread. It's almost globally distributed, with the exception of polar systems, which means that there are lots of different natural patterns in the thermal environment that we're able to take advantage of. They're abundant, and most importantly, they're lab cultivable, right? So we can go out and collect these copepods from the fields, bring them back to the lab, and then raise sort of hundreds of thousands of them for multiple generations. And so that's what my dissertation was about, was looking at patterns in the evolution of thermal tolerance and plasticity and thermal tolerance over spatial and temporal scales. So I'm going to take a really macro level perspective on these projects, since most of them are published now, and instead try to synthesize across these projects to get at one key point. But I do want to introduce the studies that I'll be talking about, that I'll be pulling data from. A couple of my projects looked at these spatial patterns and the evolution of tolerance and plasticity, taking advantage of this really large latitudinal temperature gradient that we get along the east coast of the US, from Florida up to Canada. We were also able to take advantage of several different temporal patterns. So for example, the waters in coastal Connecticut in Long Island Sound have an annual temperature range of over 25 degrees Celsius, which is similar in magnitude to what we see across this latitudinal temperature gradient. And so the question is, for copepods that have a generation time of about a month, how does this relatively extreme annual temperature cycle affect the evolution of tolerance and plasticity? And then finally, we had this extensive experimental evolution project that I ran, where we took 3 populations from across this latitudinal temperature gradient, brought them back to the lab and exposed them to sublethal but chronic temperature selection, and then tracked the evolution of tolerance and plasticity across 80 generations in the lab. And so I'm going to spare you all the nitty-gritty details about what we found for each of these projects, and instead try to synthesize across everything. And we can do that because each of these projects included a component that had the same experimental design, right? We essentially repeated the same experiment across these latitudinal and temporal scales. And so we have a general schematic of what it is that we did. We would take copepods from the fields, bring them back to the lab, raise them for a couple of generations to minimize the effects of previous environmental acclimation. And then we ran them through this split brood common garden experiment, where we'd take eggs, split them into 2 groups, and then raise those groups at different temperatures, either 18 degrees for a sort of cool temperature, and then a warmer temperature sort of between 22 and 24 degrees, depending on the experiment. And this split brood design allows us to partition the effects of sort of genetic differentiation, plasticity, and variation in plasticity on thermal tolerance itself in these copepods. And so to measure thermal tolerance, we waited till the individuals were mature, exposed individual copepods to an acute heat shock at a range of temperatures, and then used that sort of binary survivorship data to estimate these thermal survivorship curves. And I am going to show all the curves just so we have some idea about what it is that we generated. Across all these projects, we ended up with 93 survivorship curves using just under 20,000 individual copepods, or 20,000 individual measurements of copepod survivorship. And just from this sort of broad perspective, we can see that there's a lot of variation in these survivorship curves. And again, because of the experimental design that we used, this variation can be partitioned into the effects of genetic differentiation, of thermal tolerance, and plasticity. There's one last sort of quantitative step that we have to take, though, to talk about what I want to talk about today, which is sort of reducing each of these curves to a single number so that we can estimate plasticity easily. This is an example of the survivorship curves from one of our populations in the latitudinal studies. And so for each of these curves, we estimated thermal tolerance as LD50, which is the value, the temperature that we'd expect, 50% survivorship. And then, so by estimating thermal tolerance in this way for both developmental temperatures, we can get a value called delta LD50, the effect of sort of developmental temperature on thermal tolerance. This is our metric for plasticity. And then finally, to synthesize across these projects, because there was slight variation in the upper acclimation temperature that we used, we standardized everything as an acclimation response ratio, where we divided delta LD50 by the difference in developmental temperature. And so the metric for plasticity that I'm going to talk about today are these acclimation response ratio values, which are essentially the change in thermal tolerance per degree difference in the developmental environment. Okay, so using this experimental design, we were able to go through and measure thermal tolerance and plasticity in thermal tolerance for a number of populations, for various seasonal collections here in Long Island Sound, and then for multiple generations throughout this experimental evolution project. And we were then interested in taking this data on plasticity and asking, can the patterns in the evolution of plasticity that we're seeing be explained by some of those hypotheses that I mentioned at the beginning, the climate variability hypothesis, which relates ARR values to the variability in the thermal environment, or are they sort of better explained by patterns in thermal tolerance? And so we're going to start off looking at the patterns with the climate variability hypothesis. We have the acclimation response ratios on the y-axis plotted against temperature range. This is either sort of the absolute temperature range experienced during the season of occurrence for our latitudinal populations, or the temperature range experienced by the F0 generation, so the copepods that I collected for our seasonal collections, which we were able to measure because we have access to this really incredible continuous temperature data set for our collection site. And what we see is that for all of these, the experimental evolution data is excluded from this figure because temperature range is 0 in the lab, so it doesn't really tell us all that much in this form. But for the other 3 projects, what we can see is that there's positive but weak and non-significant relationships between plasticity and variation in the thermal environment. And this does hold for other forms of plasticity, or for other forms of variation, but we are sort of limited in the metrics we can use by what we can pull out of our temperature data sets. But sort of no matter which way you slice it, we don't have strong support for the climate variability hypothesis for our species of copepod. We can contrast that with what we see for what we see with patterns in the evolution of plasticity against thermal tolerance, which is shown here, using the lower acclimation temperature as our sort of baseline. And in this case, for all 4 projects, including the data from the experimental evolution project, we see the strong negative and consistent relationship between the evolution of plasticity and evolution of thermal tolerance, which does provide support for the trade-off hypothesis. I want to note really quickly, though, that sort of we call it the trade-off hypothesis, but there are multiple processes other than a true mechanistic trade-off between these between plasticity and tolerance that can create this negative relationship. But so sort of as a matter of convenience, we call it the trade-off hypothesis. But anyways, regardless of what's creating this pattern, it's interesting that we see a consistent evolution of plasticity, a consistent pattern across both spatial and temporal scales. So to sum up, across these different projects, we found evidence for genetic variation in both tolerance and plasticity, either between populations or between collections across generations. This is nothing new. This adds to a large body of literature that shows the same things. But what we were able to do is take that data and then show that we see this, these concordant patterns in the evolution of plasticity across these different scales, following this negative relationship between plasticity and thermal tolerance. I mean, this has a couple of interesting implications. One, it provides a nice source of sort of anecdotal evidence that these space for time substitutions that are common in comparative physiology and ecophysiology might actually yield relevant information about how populations might respond to environmental change. But maybe more interestingly, in my opinion, is this idea that adaptation to future warming, right, through the increase in thermal tolerance might not actually reduce vulnerability to climate change if it comes at the expense of plasticity, right? Plasticity is an important mechanism that helps populations cope with variation in the thermal environment. And climate change is not only producing warmer weather, but more variable climates. And so this loss of plasticity might hinder populations in the future, supporting this idea that maybe we should be looking at within species patterns in plasticity. So with that, I want to thank a couple people before I take questions. Carl Schlichting was on my committee, and so he obviously played a large role in how I think about the evolution of plasticity. And then I also want to thank the awesome undergrads who helped out with several of these projects over the last couple of years, as well as our funding sources. And so with that, I think we have time for questions.
Great, thank you so much, Matthew.
Yeah, thank you, Matt. We have a question in the Zoom window. Do any of your data hint at the underlying cause of the negative relationship between tolerance and plasticity? You mentioned it might not be a direct mechanistic trade-off.
Yeah, that is sort of the million-dollar question with this data. I think that one of the best explanations actually isn't a true mechanistic trade-off, but instead that there are these hard upper thermal limits that sort of nothing is able to overcome, no mechanisms are able to overcome for this system. And so that as you approach that upper thermal limit, the ability of plasticity to affect thermal tolerance further is diminished, right? You sort of hit a wall. And so that would produce the negative pattern that we see, which I think is probably what's going on.
Awesome. So we have another question in the chat. What was the response ratio a ratio of?
So the response ratio is for the change in thermal tolerance per degree difference in developmental temperature. So what's the effect of developmental temperature on thermal limits? I blasted through those methods really quick. So
Always good to clarify. So feel free to ask more questions in the chat. I think people can't message everyone. They can just message me directly. So feel free to do that either in the Zoom window chat or on Slack. I had a question just as we're, and this is my ignorance about copepods and the species in particular, but the species is a huge range. What kind of population genetic structure is there across, I guess, its global distribution?
Yeah, so that's one of my chapters was looking at sort of how does population structure play into these patterns that we see. And the simple answer is that it's a nightmare with Acartiatansa specifically. It might be one of like the worst systems out there because we end up with sort of really deep cryptic divergences that are sometimes allopatric, sometimes they're sympatric. And so it's this mix of genetic differentiation and cryptic speciation and then sometimes you get massive amounts of gene flow, right? So we find the same haplotypes in Florida and in Canada, and then sometimes they're really different in just over 10 kilometers. But it does all come together in a nice story about how gene flow constrains the evolution of tolerance and plasticity.
Awesome, thanks. So there's one more question from Joey Bernhardt. So first off, awesome talk. Did you observe a latitudinal gradient in the thermal tolerances, or what do you think drives the intra-specific variation in thermal tolerance?
Yeah, so there is a nice correlation between thermal tolerance and mean temperature, mean or maximum temperature, because they're also strongly correlated. And so we think that's what drives these patterns in thermal tolerance, which in turn drive patterns in the evolution of plasticity. That was a good question.
And Joey says, thank you for your response. And if questions come in after we run out of time, I'll just post them to the speaker, and all the speakers are co-hosts, so you can respond to them in the broader chat window.
Awesome. Thank you.
Thanks, Matt.
Okay, so to keep on schedule, thank you so much, Matthew. Let's move on to our next talk. Our next speaker is Sam McKlin, and they'll be speaking to us. Oh, that just took away my schedule. They'll be speaking to us today about assessing the feasibility of assisted gene flow as a conservation strategy to mitigate the effects of climate change. Take it away.
Hello, my name is Sam McKlin, and I'm a senior at Davidson College in North Carolina. And today I will be talking about the feasibility of assisted gene flow as a conservation strategy to mitigate the effects of climate change. So climate change will negatively affect a diverse array of species as the occurrence of extreme events increases and precipitation and temperature regimes are altered. It has been shown that 1 in 6 plant species are threatened by our current climate trajectory. And more specifically, we're seeing that maladaptation is becoming more widespread. Range sizes are shifting, so species are moving polewards and upwards in elevation. And we're also seeing extinction risks that are increasing. And so conservation practitioners are looking for ways to alleviate these threats. And one specific conservation strategy is called assisted gene flow. And so I'm going to walk through a quick example of how assisted gene flow works in theory. So say we have 2 populations where all these circles represent individuals, and the color of those individuals represents the environment that they are their ideal environment, the environment that they're locally adapted to. And the colored bar below the populations represents their actual physical environment. So you can see the left population is locally adapted to its physical environment, but the right population is not. And so with assisted gene flow, we can identify populations as targets, like the one on the right, that could benefit from a source population, like the one on the left, that is adapted to similar conditions to the right population's physical environment. And so with assisted gene flow, we can physically transport individuals from this population, from the source, and put them into the target population. And so initially we might see some kind of demographic rescue in the target population as population size increases and genetic variation expands. And over time, we might see introgression start to begin, individuals start to hybridize, we see more genetic variation until eventually we have a response to selection and adaptive evolution. And so in reality, there are multiple potential sources that are all adapted to their own, locally adapted to their own physical environments, all located at different distances from whatever potential target population we're talking about. And so the question becomes, which source population is the best? And also, would climate change affect that choice? So if you're trying to make an assisted gene flow program targeted at a future environment's physical conditions, what would that look like? And so also, in order to assess how well an assisted gene flow program is doing, we calculate a population growth rate. And in order to do that, we break down the study species life cycle into several fitness components that are listed here as a couple examples, like survival and growth. And that brings up the question, which fitness components determine the success of an assisted gene flow program? So which of these fitness components might be potentially limiting population growth rate and would be important to focus monitoring efforts on? And so we pose a couple of questions. First, which source population do we pick, which is ideal for an assisted gene flow program? Second, does the ideal source population change if you're targeting future climatic conditions? And third, which fitness components most influence population growth rate? And so in order to answer these questions, we implemented a 3-year assisted gene flow program with the perennial plant Bucra stricta, a wild mustard plant, in a climate manipulation experiment in the Rocky Mountains at Rocky Mountain Biological Laboratory. And the experiment took place between 2016 and 2018, and we analyzed the data this past year. And so in our system, we have 5 gardens here positioned along an elevational gradient. And there's a locally adapted Bucra stricta population at each of these gardens. And what we did was we reciprocally transplanted individuals from each population into each other garden. And we did this for all of the gardens so that eventually we have all 5 gardens represented at each garden, including the local population to that garden. And so in this system, we observed 6,703 rosettes and almost 12,000 seeds. And we use a subset of these data to construct assisted gene flow scenarios that I will show you shortly. Additionally, there are 125 genotypes represented here with an average of about 25 locally adapted to one of the 5 gardens. And so here is a picture of the highest elevation garden, and you can see it is a much more green and lush environment than the lowest elevation garden, which is located about 800 meters further down the mountain. And also at each garden, we imposed, on half of the individuals at each garden, we imposed a snow removal treatment that simulates future climatic conditions. And so this exposes individuals to reduced snowpack, it reduces water availability, and exposes the individuals to sub-zero or more variable air temperatures, which in turn makes them susceptible to frost. And these are all conditions that we would expect to happen under climate change. And the other half of individuals at each garden just experienced ambient snowpack, so the present climate. And so for our assisted gene flow scenarios, we chose to use the top 2 locally adapted populations in the top 2 gardens as target populations for our assisted gene flow program. So the idea is that if conditions are becoming warmer and drier with climate change, genotypes from warmer and drier adapted populations could help boost population growth in these higher elevation gardens. And so we constructed these scenarios that you see here. The X's, the white X's are the target populations, and the solid circles are the source populations. And so you can see that it's the highest elevation garden supplemented by the lowest, second lowest, third, fourth, and so on. And then also the target populations on their own. And so we actually have twice this many scenarios because of the snow removal treatment. So at each garden, for each scenario, we have all of these scenarios at the present climate and then the simulated future climate. And so we also have several, well, so this is a life cycle diagram of Bucra stricta. And there are several fitness components that you see here. And we collected data on all of these fitness components and then used mixed models to model those fitness components. And then we took those model outputs and put them in an integral projection model in order to estimate a population growth rate. And so the integral projection model takes a population of size n with a size distribution z at time t, and multiplies that by the sum of these two routes through the life cycle, with P being the adult survival and growth route, and F being the reproduction fecundity seedling to rosette route. And then we bootstrapped each integral projection model 2,000 times to obtain 95% confidence intervals around our estimates. And so for the scenarios, we first estimated population growth rate for the target populations on their own. And then for the actual assisted gene flow scenarios, we pooled fitness component data for the target population and the source population and treated that as one population that we estimated a growth rate for. And we did this for each scenario. And specifically, we predicted that transplants from the next lowest garden would best improve population growth rate because they're in environments that are most climatically similar to the target population. And we also predicted that under the snow removal treatment, transplants from further down the mountain than the next lowest garden would best improve population growth rate. Because those sources experience warmer and drier conditions in their home sites that might prime them for the simulated future climate that they'll experience in the snow removal treatment in their target site. And so we observed that in the target population for the highest elevation garden, that there was stable growth in the control and declining population in the snow removal simulated future climate. And then when we supplemented that target population with 4 other sources, we found that in the next lowest garden, that scenario, we got a boosted population growth rate in both the control and the snow removal treatment. And we did not see that assisted gene flow benefit in the other 3, from the other 3 sources. And for the second highest garden as the target population, we observed a stable growth in both the control and snow removal on their own. And then we did not observe an increase in growth rate when we supplemented that second highest population with other sources. In fact, we saw a decrease here. So assisted gene flow is not either neutral or beneficial, but it can also be harmful to population growth rate. Additionally, we estimated fitness component elasticities. And so an elasticity is the fractional change in population growth rate that we see when we perturb an individual fitness component at all size classes. And so basically that means that the higher the elasticity, the larger effect that changes to an individual fitness component will have on the overall population growth rate. And so these are the elasticities for the highest elevation population on its own. And we see that adult survival and growth have the largest effect on population growth rate. And when we look at the one assisted gene flow scenario that boosted population growth rates, we see that recruitment got a little bit higher, so seedling recruitment into adults, but across the board, adult survival and growth are still the most important, the most influential on population growth rate. And so to quickly recap, which source population do we pick? We have evidence that the next lowest elevation population, even under snow removal, boosts population growth rate, at least in this one scenario. But beware, supplementing populations can also reduce population growth rate. And then next, does the ideal source population change if you're targeting future climatic conditions? We did not observe that in this study, as the assisted gene flow scenario that worked also worked in both control and the snow removal. But perhaps this could be a factor in other study systems. And lastly, which fitness components most influence population growth rate? We saw that it was across the board, adult survival and growth. And so that's important for conservation practitioners to know, so that they can focus monitoring efforts on adult survival and growth, so on adult rosettes. And so I want to thank my supervisor, Dr. Susanna Vadimar, and our collaborator, Dr. Jill Anderson, the National Science Foundation, and Rocky Mountain Biological Laboratory, for without whom this project would not have come together. And also, I am looking for lab tech and post-bac positions in computational biology and data science. And you can always reach me at samacklin@davidson.edu, and I am happy to take questions.
Great, thanks, Sam. So if you have questions for Sam, either send them to me in the Zoom chat or post them on Slack, on the Climate Slack channel. Great talk. Well, while we're waiting for questions, I'll ask one that I was thinking about. In the populations that do better when you supplement them with the next lowest population, is the growth because those individuals are doing well there, or have you quantified gene flow to see if you're like getting admixed individuals?
Yeah, so we haven't, I kind of forgot to mention this in my talk, but we didn't look at the hybridization yet of these populations. So the whole thing was that our population growth rate estimates are, I'm kind of looking at that initial stage of that initial demographic rescue that you would see from assisted gene flow. But yeah.
Okay, awesome, thanks. So there's another question from Sam Shiner. Can you predict which source population is likely to provide a benefit based on patterns of adaptation?
Sorry, can you repeat that?
Yep. Can you predict which source population is likely to provide a benefit based on patterns of adaptation?
So we're looking at so our study system showed widespread local maladaptation. And we observed this in a previous paper.
Okay, thanks. I have another one here from Anna Hairgraves. Did population growth rate integrate across all source populations, i.e. one might expect an initial decline in fitness while selection weeds out the maladapted genes, then an overall increase?
Sorry, I'm a little bit off my
It's
game.
okay, I can send it so you can see it.
Yeah, sure, that would be helpful, thank you.
Actually, you can't really copy in these. I'll just repeat it. We have some time. Did population growth rate integrate across all source populations? I.e., so in other words, one might expect an initial decline in fitness while selection weeds out the maladapted genes, then an overall increase.
Yeah, so we think that will likely happen over time, yes.
Okay, and I'll ask one more. Why do you think that you saw a negative impact of artificial gene flow in your study populations?
Yeah, so that's a really good question. The thing is, some of these the source populations were very maladapted to their home sites, and so since we see widespread maladaptation across our entire study system, yeah, so it's basically local adaptation is the root cause there.
Awesome. Do we have time for one more? What do you think? One more. Okay, so this one's in the Slack channel. So if you don't get to completely answer it, just visit that. It's from Joey. Thanks again for a great talk. I'm curious, you have any thoughts on what the costs of having a high thermal tolerance might be? I'll just leave it there. There's another part of it, but you can answer that. Oh, sorry, this is for a different talk. I'm doing a bad job. So that's that's for Matt. Matt needs to go into that in the Slack channel. And I think with that, we should probably start to transition. So thanks again for a great talk, Sam, and sorry that I'm bad at doing this. I'll try to be better.
I can continue to answer stuff in Slack if you have other things.
Thanks, Scott. And you're not doing a bad job. You're doing a great job. And that was a great talk, Sam. Thank you. So let's move on to our next speaker. Our next speaker is Matias. Oh, every time the speaker comes up, my screen shifts. So Matias Gomez-Corrales, and he will be speaking about cryptic lineages matter for coral conservation under climate change. Take it away.
Hello, everyone. Thanks for coming to my talk, at least virtually. My name is Matias Gomez. I am a second-year PhD student in Dr. Carlos Prada's lab at the University of Rhode Island. And today I'm going to talk about cryptic lineages and coral conservation. So we know that climate change, mainly through the process of global warming, causes coral bleaching, as we can see here on the graph on the right. Temperature anomalies have been steadily increasing over the last century, which has created the breakdown of the coral-zooxanthellae symbiotic relationship, which in turn has created coral cover decline, mainly because by having coral bleaching, the colonies or species that experience it end up having a high degree of tissue mortality, which has created a global trend of coral cover decline, as exemplified here on the graph on the right, in which we have 40 years of coral cover measures, and we can see the actual trend of coral cover decline for the Caribbean in this case. So how coral reefs will respond to this ongoing thermal stress will have to do with the distribution of genetic variation that you find within and among species. And this is important because those species may differ physiologically, which brings me to the importance of doing an accurate species delimitation process to be able to spot the different physiological thresholds species may have. So as we can see here on the diagram I made, on the right, we have a group of 3 species, and they all have different ranges of heat tolerance. Alternatively, another scenario would be in which the physiologic or the genetic variation that underlies physiological thresholds would be randomly distributed among these 3 different species, in which case this diagram won't hold true. So talking about coral species boundaries, these have been very hard to establish due to different biological processes, such as introgression or hybridization, which confound the phylogenetic signal when you are doing phylogenetic reconstructions. Also, coral species have a great deal of phenotypic plasticity, meaning that you can have the same species with a great degree of morphological variation just because they are found in different environments. Also, there are cases in which recent speciation events make more difficult to disentangle those recent speciation events from all divergent events. And finally, we have different mutation rates of the nuclear markers when compared to mitochondrial markers. So this brings me to the topic of cryptic species and why they are hard to spot or why they sometimes remain elusive to science. So we refer to cryptic species as those genetically distinct but morphologically similar species. And as we can see here on the diagram on the right, we have a group of black stars seemingly alike. However, if we take a closer look, we can see that there are 2 different groups that are both interconnected by blue lines, and reproduction is not possible between these two groups just because they have different genetic makeups. So this is a diagram to illustrate the concept of cryptic species, and this has more importance when these two groups have, for instance, different or harbor potential physiological variation to withstand different environmental stress. So taken together, the importance of delineating cryptic species under a climate change scenario must be a research priority, because this helps us understand current ecosystem dynamics as well as forecasting future ecosystem or future reef states or ecosystem changes. And at the same time, they'll help us understand whether or not the changes are being driven by few species that have a wide distribution range, or by actually multiple species that have a more limited distribution range. So this brings me to my study system, which is one of the major reef builders in the Caribbean. This is the mountainous star coral, Orbicella fabulata. This is a massive, shallow species, widely distributed along Caribbean reefs. It is part of a species complex with another two species, and is considered currently as a threatened species by the Environmental Species Act. So, leveraging genomic resources available for this species, we use the Orbicella fabulata genome, and we relied on data from a recent publication in Molecular Ecology, in which the authors scored quantitative genetic variation, as well as experimental thermal stress challenges from colonies of Orbicella fabulata found in Bocas del Toro, Panama reefs. So regarding the main finding that the authors found in this study was that they were able to find heat-tolerant colonies within the colonies that they sampled in these reefs in Panama. And the way they were able to find these colonies was by establishing this bleaching score, in which they measured the increase or decrease or the change in the symbiotic algae present in the coral's tissue before and after exposing the colonies to thermal stress challenges. So finally, they found a few colonies that were able to withstand, or that were tolerant to bleaching, which led them to conclude that there is substantial genetic variation to withstand warming oceans within this species in the area under study. However, given the complex evolutionary history of corals and the recent coral speciation events in the Caribbean, we suspected that the physiological differences or the physiological responses to bleaching could be related to cryptic lineages. And this is the reason why we ask ourselves a series of questions. The first one is whether or not Orbicella fabulata is a single biological species. And if it's not, is there any evidence of cryptic lineages within the genetic data that we reanalyzed? And if there is actually evidence for cryptic lineages, do they differ in their bleaching susceptibility? So to answer this series of questions, we relied on genomic data, originally from 43 colonies, but after removing clones, we ended up having 39 colonies or individuals, from which we scored 3,560 high confidence SNPs. And we took a population genomic approach based on multivariate analysis and clustering methods, as well as a phylogenomic approach based on maximum likelihood and coalescent-based methods. And finally, to study the symbiotic community present in these colonies, we used the ITS2 marker to do taxa prediction of the symbionts present in the symbiotic community. So our main finding is that there are actually 3 cryptic lineages hidden within the genetic data that we reanalyzed, as we can see here on the discriminant analysis of principal component graph on the left. We found 3 distinct genetic clusters. These 3 groupings were further recovered by the neighbor joining tree. as well as the Bayesian clustering algorithm of a structure. As we can see here on the right, we found at least 2 main groups, with a third group nested within the largest group in red. So we found a third group in green, in this case, nested within the largest group, the red one. Regarding the phylogenomic methods that we used here, our maximum likelihood tree recovered the same topology as the neighbor joining tree with a good bootstrap support for most of the clades. And then we use the package snap within the program BEAST to test for the most likely number of species present in the data, for which we got the strongest support for 3 species that we can see here on the right in the maximum clade credibility of a snap. That represents the species in thick blue. Talking about the symbiotic taxa present in the colonies that we reanalyzed, we found that the symbiotic community was dominated by 4 genera. However, when we mapped the taxa identity of the symbionts against the cryptic lineages that we uncovered in this study, we didn't find a one-to-one relationship, meaning that there is a high degree of flexibility or heterogeneity in the relationship between the cryptic lineages and the symbiotic taxa, which led us to our main conclusion that the cryptic lineages that we found here actually responded differently to coral bleaching. And we can see that here in the bleaching score, in which we have colonies labeled color by cryptic lineage, and we can see that most colonies that were tolerant belong to the red lineage, while only 1 blue colony was tolerant to bleaching. And the remaining cryptic lineage in green only had colonies susceptible to coral bleaching. So we conclude that we were able to find cryptic species under the nominal species of Orbicella fabulata in Bocas del Toro reefs in Panama. Most importantly, these cryptic lineages differ in their bleaching susceptibility, which taken together means that the Orbicella fabulata conservation status is actually more acute than we previously thought. And with that, I'd like to thank you all for your attention. I'd like to thank the organizers of the conference, my advisor, and also all the institutions that made this research possible. And with that, I'll be happy to take any questions. Thank you so much.
Great, we have plenty of time for questions, so feel free to send them to me in the chat and I will read the right ones, hopefully. Awesome, so we have a question from Jen. Great talk. Do you know how these lineages are distributed across the landscape? Is there any evidence for reproductive isolation?
Hi, thank you. That's a great question. I'm not entirely sure regarding this data. I'm analyzing some other data from other areas in the Caribbean. We suspect that there might be a depth climb, segregation across depths. But I'm not entirely sure how that genetic variation or lineages are distributed across the landscape. And I think there was something regarding about reproductive isolation. So we haven't tested that yet. We have been studying just the genetic variation, and we wanted to couple that genetic variation with the bleaching score that was published in this article.
I have a question while we're waiting for others to come in. Is the red lineage geographically more widespread than the green or blue lineages on that distribution map?
Well, right now we only analyzed, the data that are presented is restricted to a few reefs in Panama in a kind of limited geographic range. But that cryptic lineage seems to be all over the reefs that we sample. And currently I'm working on other data from other parts of the Caribbean in which we have spotted some also cryptic diversity, and we're still working on that to actually see if what we've seen in different areas is actually the same cryptic lineage or not.
Great. We have another question. How do you normally distinguish coral lineages? And how are species generally identified in corals? And how does this influence their conservation?
That is a great question, because typically, at least for the last 40, 50 years, we relied on morphological characters, which are actually highly variable. And nowadays, since the start of the genomic revolution, we'd be using single genes, but there are some issues with the genes that have been used for phylogenetic reconstructions in corals. So right now, what I'm showing here is not completely new. We suspected previously, or many other researchers have suspected previously, that there might be more variation in the number of species that we see all over the reefs around the world. So right now, using SNPs or using UCEs, we are doing more phylogenetic reconstructions because morphology is very hard to interpret in order to do the lineage or the species delimitation.
Great, we still have about 5 minutes for questions. So if other people have questions about corals, feel free to send them my way. Great, we have another question in the chat. Nice talk. Any plans to do a GWAS to identify potential variants related to heat tolerance that vary among lineages?
That is actually something that I've been considering, and it's part of another chapter of my dissertation. I like to do some acute thermal stress challenges, previously identifying the cryptic lineages so we can actually have a scope of all the variation that we have and then subject the different cryptic lineages to thermal stress challenges. So we can do some GWAS analysis and kind of tease out the variation related to bleaching or resistance to bleaching.
Awesome. I have a question because I don't know anything about corals. Does this species have coordinated spawning? Do all corals do that? Like across the Caribbean, or no? I don't.
That is partly true. I mean, some of the broadcaster species or the broadcast spawner species, they tend to be very coordinated and well synchronized in the reproduction efforts. Sometimes there are differences in the latitudinal or the geographic latitude in which you sample corals. And some other evidence sometimes comes from asynchronous spawning. So sometimes you have, or some cases in the Pacific reefs, they found that they were able to spot the cryptic lineages or different species just based on the time of their spawning.
Great. We have a couple more questions coming in. Are there other tolerances to is there other tolerances to other abiotic factors that can give us more information about lineages? So are there other abiotic factors that you might be able to use to distinguish lineages from one another?
Right now, we only, I mean, the data that we reanalyzed was only kind of coming from shallow depths. But when we have a more distributed kind of colonies in a deeper environment, sometimes you can use light, or we can use also the respiration rate of different colonies when you subject them to oxygen measures. So sometimes you can use the respiration rate or also the light levels at which they're exposed at different depths.
Great, we just have a couple more minutes.
Yeah, if anyone wants to ask something else, they can just Slack me or post it somewhere else.
Yep, and if anything comes up in Zoom, I'll send it directly to you.
That is great. Yeah, thank you so much.
Thanks for a great talk.
Okay, thanks, Scott, and thanks, Matias, for that great talk. It's about time for us to move on to the next talk. So our next speaker is Samantha Watson, and she'll be speaking about drought effects on plant functional traits in California native grasses.
Hello, my name is Samantha Watson, and I am a master's degree candidate at California State University, Los Angeles. Today, I am presenting functional trait response to drought in California native grasslands. Man-made climate change is affecting temperatures and precipitation regimes globally. Over the last century, average temperatures have increased 1.6 degrees. And the projections predict that this trend will continue for the next century. This map shows how global temperatures are projected to change. The map takes predicted temperatures for 2099 and subtracts temperatures experienced in 1900. We can see that much of the map is within the range of a 2 to 6 degree increase. Drought and flood are also expected to increase in duration and frequency. Some places are expected to be affected more than others. This second map shows a change in precipitation from 1900 to what's projected in 2099. There are places in blue that will have more precipitation, and others, including here in California, that will become drier, shown in brown. Mediterranean climates are expected to be affected by increases in temperature and drought due to already arid climates. The map here shows the various climate regions of California, and as you can see, the climate of the greater Los Angeles area varies from Mediterranean to arid, and as such is expected to experience an increase in the frequency and duration of drought. So why should we care about increases in temperature and drought? There are plenty of studies that demonstrate how drought affects ecosystems. For example, Penuelos et al. 2007 measured plant growth using above ground biomass among 3 herbaceous species in a Mediterranean climate. The graph displays air temperature on the x-axis and above ground biomass on the y. We can see a decrease in biomass with increasing air temperature. The second panel of this graph displayed soil moisture on the x-axis and shows that above ground biomass decreases with decreasing soil moisture. So both increased temperature and decreased soil moisture reduce biomass, and together they can combine to further negatively impact growth of many species. So how does increased temperature and decreased soil moisture affect plant growth? Some basic plant physiology can help us better understand. Soil moisture enters through the plants, through the roots, and then travels up the xylem to the leaves. Plants need this water in their leaves for photosynthesis. Plants also need to open their stomates in their leaves in order to take in CO2. When opening stomates, plants risk losing some of their water, and this risk increases when the air around the plant is dry. With increased atmospheric moisture, the plant can maintain more of its water. This is due to vapor pressure deficit, or VPD, the difference between the amount of water the air can hold and the actual water content in the air. During drought, there is an increase in the water demand on a plant due to loss of soil moisture, as well as the decreased atmospheric moisture. And this can cause a plant to change its strategies for maintaining its water. One strategy is to reduce water loss through its leaves by altering its morphology. Changing its specific leaf area, or SLA, the plant can preserve water. Specific leaf area is the area of a leaf compared to its mass. In other words, a large wide leaf that is thin will have a greater SLA, resulting in greater water loss than a thicker leaf with a smaller surface area. In addition to altered SLA, a plant may also have a different strategy for drought conditions. A plant may alter allocation of energy towards growth of roots in an attempt to gain access to more water. Altering the ratio of fine and coarse root production is one way a plant can attempt to gain more water. Fine roots is where a plant can access soil water. So if a plant increases its fine root production, it can increase its chances of gaining water. Coarse roots are for transport of water, and a plant can save energy by reducing the amount of coarse roots it produces. In addition to strategies for increased water, the diversity of a plant community can also affect productivity. Increased biodiversity can mitigate the negative effects of drought on a community, and individuals in a community can vary their use of available resources. And a diverse community of plants can also alter the microclimate. Another way biodiversity affects the plant community is drought-sensitive species can be buffered from negative effects with increased diversity, as seen in the stress gradient hypothesis. On the x-axis here, we have an increase in precipitation, and on the y, we have the drought-sensitive species performance. In the first graph, we see that a drought-sensitive species is greatly affected by changes in precipitation, both in the negative direction with little precipitation and positively with increased precipitation. As we increase the number of species, we see that the drought-sensitive species is less affected by changes in precipitation, to the point that there is no effect of precipitation on the drought-sensitive species. The effect that we see could be due to microclimate amelioration and altering of the microclimate by other species, but this could also be due to decreased intraspecific competition. Knowing that in the future, increasing temperature and decreasing precipitation will affect plant growth, it's important to understand the extent of these effects. Climate studies should strive to replicate drought conditions. Currently, climate studies looking at drought erect rainout shelters, and these rainout shelters stop direct rainfall from landing on the soil. The problem with these studies is that they do not take into consideration humidity in the atmosphere. Rainout shelters have been shown to produce unwanted artifacts, including accumulated humidity under the structures. Another problem for these studies using rainout shelters is that they fail to address the decrease in humidity that we would see in a natural drought. From a study done in my lab, Aguirre et al., we looked at plant growth using total above ground biomass. We show that there is no change in above ground biomass between dry soil and wet soil when the air is humid. There is, however, a change in biomass when the soil and air are both dry, like you would expect to see in a true drought. For our current study, we came up with 3 hypotheses. First is that a model species will respond to drought by reducing SLA. This will be the strongest when drought combines soil drought and atmospheric drought, and that this will also be strongest when grown alone without microclimate amelioration effects that a higher diversity mixture can provide. Our second hypothesis is a model species will have greater allocation to fine root production relative to its coarse root production when grown in drought conditions. This will be strongest when drought combines soil drought and atmospheric drought. And fine root production will be highest in polycultures where competition for soil moisture is high. And then our last hypothesis is that a model species grown in dry soil and dry air, like a true drought, will have a different growth strategy than that of the same species grown in dry soil and humid air, similar to the current drought experiment conditions. And for our methods, our field site is on the California State University campus here in Los Angeles. We planted grasses into 32 mesocosms, and we used 2 levels of soil moisture, 2 levels of humidity, and 2 more levels of diversity for a total of 8 different treatments. Our diversity levels include an 8-species polyculture of California native perennial grasses, including Poa secunda. And our monocultures are all Poa secunda, which we used as a phytometer to measure the physiological responses from the environment in our gradient of drought conditions. Our first level of treatment is wet soil and humid air. We added humidity to the air of our mesocosm using commercial grade humidifiers and added water to the soil based on the 50-year average in Los Angeles, California. This one is our wet or mesic condition treatment. Our second level is dry soil humid air, where we added the commercial grade humidity to each of our mesocosms, but we reduced the amount of soil moisture added to each pot. This one would be similar to what you would find in current drought experiments where they use rainout shelters. And our third level is wet soil dry air, where we used the 50-year average of California, of Los Angeles, California, and we left the air California ambient, which, as you know, is dry and arid. So we have the wet soil and dry air resulting in our California ambient control. Our fourth level is dry soil, dry air, where we reduced the amount of soil moisture, and we also left the air to be California ambient. This one was to represent a true drought. So for our data collection, we chose the two largest individuals of Poa secunda from each pot to a total of 64 individuals. These individuals were measured for their leaf mass and also for their area using ImageJ software. We also took above ground biomass. We took half of the biomass on March 19th, and then the remaining biomass was all harvested on May 13th. In addition to the above ground biomass, we took soil cores, which we separated into two measurements. From the top of the soil and then down 16.5 centimeters, and then from 16.5 centimeters down to 33 centimeters, which was the bottom of our pots. These soil cores were used in order to gain roots. We took the soil cores and we rinsed and cleaned them using mesh sieves to extract the roots. The roots were then separated into fine and coarse depending on their diameter. And then they were dried and weighed. So now to our results. So for our first hypothesis, a model species will respond to drought by reducing SLA. We found that there was no significant difference in SLA between treatment groups, but we did find that our monoculture leaves were thicker with less surface area when compared to our Poa secunda grown in polycultures. This may be due to microclimate amelioration in our polycultures or possibly from alleviated intraspecific competition. For our second hypothesis, looking at a model species will have greater allocation to fine root production when grown in drought conditions. We first looked at our overall root mass to try to fully understand what's happening with our roots totally. So with keeping in mind that all of our polyculture root data includes all species in the mixture because we did not have the ability to separate them out. Our monocultures, we see the expected increase in root production when comparing wet and dry soils. So in the dry soils, we see that there's more root production in an attempt to gain more access to water. When we look at our polycultures, however, we see this unexpected result with a decrease in the root production of humid air when comparing wet and dry soils. Looking at our shallow fine roots, wanting to see what we find significant. We see that there is less root production in our monocultures with humid air and wet soil, showing that there is plenty of available soil moisture for our roots to access. And all three of the other conditions in our monocultures, there is an increase in root production. Our polycultures, again, we also see this decrease in root production of dry soils when looking at our humid air polycultures. One hypothesis for this that we came up with is that the grasses may be accessing water through accumulation of water on their leaves rather than through the soil, since they have plenty water above ground and do not have enough below ground. When looking at our shallow coarse roots, we see that polycultures grown in humid, wet conditions, they're producing more coarse roots than any other condition. We also see that both monoculture and polyculture in dry air result in an increase in root production when the soil is also dry. So there is a greater demand on the dry soil, dry air, and therefore increasing their root growth. For our third hypothesis, that a model species grown in dry soil and dry air will have a different growth strategy than the same species grown in dry soil and humid air, we used a principal component analysis to reduce the complexity of looking at so many different traits, to try to see if we can tweeze out any trends or patterns. And if we isolate here our dry soil and humid air, which would be similar to what we would see in our the current drought experiments, and compare that with dry soil, dry air, which would be a true drought, we can see that there is a difference of allocation of energy with very little overlap in the patterns of these two different treatments. So we found that Poa secunda did not have different SLA due to gradient changes in drought conditions, but Poa secunda did have different SLA when comparing their polycultures and monocultures. Fine root production increased in the shallow depths of monocultures when comparing wet and dry soils. We also found that polycultures had an unexpected result with decreased root production in humid air and dry soil conditions, while humid, wet soil conditions produced the most roots of all the treatments. We think that there's some further studies that need to go into this to figure out exactly why. We have that hypothesis about possibly accessing water through the leaves rather than through the soil, since the above ground moisture is plentiful. And finally, Poa secunda grown in true drought allocates energy differently than Poa secunda grown in dry soil and humid air, which would be similar to the conditions you see in current drought experiments. And with that, I'd like to thank everyone in the Wright Lab for all of your help. Thank my advisor, Sasha Wright, and my co-author, Beatrice Aguirre, and everyone else who has helped along the way. If you have any further questions that don't get answered at the end of this talk, you can email either Sasha Wright at her email, or you can email me at my email, and we're open to answering any of your questions. Thank you.
Great, thanks, Sam. So people should feel free to ask questions in the Zoom chat or on Slack. We have about 3 minutes for questions.
If anyone comes up with any questions after this, you can feel free to either email or write in the chat.
Definitely. I have a question about, oh, here, there's a better one in the chat. So do you have proposed physiological mechanisms that would allow leaves to absorb moisture in your unexpected result?
Yeah, so there's actually quite a few papers out there that talk about plants being able to absorb either dew or fog, and even rainwater. And it's called foliar water uptake. And I just started looking into it after we got our initial results. So it wasn't something that was part of our, our knowledge going into it. And so there's still more information I have to find about it. But there's a couple papers if anybody's interested in looking into. Burgess and Dawson, 2004, and Brashears et al., 2008. And there's a lot of them out there. So, yeah.
I think I'm going to cut that because now we should probably move on to our next one. Is that okay, Scott?
Yeah, that's fine, for sure.
Okay. So great talk, Sam. Thank you. Our next talk today is from Elizabeth Moore, and she'll be speaking about heat of the moment. Developmental timing of heat waves alters the outcome of host-parasitoid interactions.
All right. Hi, everybody. My name is Elizabeth Moore, and I'm going to be sharing some work I've done during my PhD at the University of North Carolina. Oops, sorry. So first off, I don't have to tell anyone in this session that climate change is affecting our global climate and the environments that we see our organisms trying to survive in. Personally, I'm really interested in parasitoid insects and their hosts. And for those of you who don't know or need a reminder, a parasitoid insect relies on an arthropod host to complete some portion of its larval development. And they inevitably kill their host at the end of development. So they play a really important role in both natural ecosystems and agriculture, serving as an important top-down control mechanism, as a lot of their hosts are insect herbivores like caterpillars. So one of the things that I'm really interested in is how extreme temperatures and especially extreme high temperatures impact these interactions. And I have a plot here on the left that's from the United States Climate Change Report from 2017, showing that the incidence of daily record high temperatures has been increasing pretty dramatically over the last couple of decades. So for this talk and for a lot of my PhD research, I have been looking into how these extreme high temperature events have direct impacts on host insects, on parasitoid insects, and then also how these temperatures have indirect effects mediated through the insects and their interaction with each other. So if you have a host that's really stressed by temperature, that can have indirect effects on the parasitoid through the host's physiology and vice versa. However, the effects of temperature don't stay the same across an organism's development. But for much of the research on thermal biology, historically, they focused on a single life stage. But as I said, different life stages have varying thermal tolerance, as we can see from on this plot on the right from Kingsolver et al. 2011. Some stages are more sensitive to temperature than others, which means that they often have lower thermal tolerances. You can see in this plot, we have eggs, larvae, and pupa of a Manduca sexta caterpillar. And you can see the eggs and pupa, their thermal performance curves are shifted to the left, meaning they can withstand less high temperatures than can larvae of the same organism. So I'm really interested in looking at these questions for a host parasitoid system. And for me, I use Manduca sexta, the tobacco hornworm, and one of its most prevalent parasitoid insects in the Southeast United States, Catesia congregata. So Manduca sexta is a major agricultural pest, feeding on such crop plants as tobacco, tomatoes, and potatoes. It's also a model organism that has been used to study insect growth, physiology, and thermal biology for most of the last century. And it's one of the most common hosts for Catesia congregata. Now, this wasp is a gregarious endoparasitoid, which are some big words that basically just mean the female wasp lays multiple eggs within her host, and those develop inside the body of their host insect. Catesia congregata is also a model system in its own right. It's been used to study the physiological impacts of parasitism on host insects for several decades. And it also has a really high impact on the tobacco hornworm, with up to 90% infection rates observed in the field. And importantly, when we're thinking about thermal biology and climate change, and for this talk, It also has a lower thermal tolerance than the host. This has been shown both by my work and a couple others, but this basically means that if you rear a parasitized Manduka at high temperatures throughout development, you see high costs for parasitoid survival and fitness at temperatures that the Manduka sexta, that when they're unparasitized, do pretty well at. So before I get into the specifics of my experiment, I'm going to give you a brief rundown into the life cycle of Catesia congregata so that you know what developmental stages I'm talking about during my experimental design and results. So we start with a female wasp looking for a suitable host, usually a first, second, or third instar Manduka sexta caterpillar. When she finds one, she oviposits into it, depositing eggs, venom, and something called a polydnavirus. These viruses are an endogenous element in the wasp's genome. They're transmitted vertically through the wasp ovaries, and they inject non-replicating virions into the host caterpillar. And these have a lot of important effects, such as suppressing host immune system early on in development, so the wasp eggs aren't killed, and later on disrupting host hormonal systems that control metamorphosis. So once these eggs hatch, we have wasp larvae that feed non-destructively on nutrients in the host hemolymph, so there's no direct tissue damage happening to the host during parasitoid development. The parasitoids go through 2 larval instars within the host, and once they are ready to emerge, they alter the feeding and locomotory behavior of the caterpillar, ceasing feeding and spontaneous locomotion. Once this has occurred, they chew their way out through the host cuticle, spin cocoons, and pupate. The host actually remains alive during wasp pupation, but the physiological changes made to it are pretty irrevocable, so that it will never eat again, and eventually succumbs to starvation, predation, or disease. So an important thing to keep in mind throughout the talk is that pretty much once you are a parasitized caterpillar, you are dead. So that brings us to my experimental question. Does the thermal tolerance of Catesia congregata depend on developmental stage? And I set about answering this question with the following experimental design. So I had 2 temperature treatments. First is the rearing treatment, 25 plus or minus 10. So we have a low of 15 degrees Celsius that ramps constantly up to the high of 35 degrees Celsius. And we've shown through prior works that this is not stressful for either the host or the parasitoid. I also had heat wave treatment, which was 31 plus or minus 11. So we had a low of 20 ramping up to a high of 42. And this heat wave consisted of 3 consecutive days. So parasitized caterpillars experienced this high 42 degrees maximum daily temperature 3 times. And this is shown to be stressful, but not lethal to unparasitized Manduka sexta. So we thought it was a good sort of baseline heat wave to use. So I took Manduka sexta eggs, reared them up to the 3rd instar, wherein I parasitized them, and then separated them into one of 4 treatments. First off, I had my control, where they just remained in the 25 plus or minus 10 rearing treatment and never experienced a heat wave. For my heat wave treatments, I had one that was early on in parasitoid development. So parasitized caterpillars entered the heat wave treatment while the parasitoids were still eggs. I had a middle heat wave where parasitoids were in the 1st instar when they experienced the heat wave. And finally, a late heat wave treatment where parasitized caterpillars entered the heat wave regime while the parasitoid were in their 2nd instar. So let's see what I found. First off, I am going to show you the results for the host caterpillars. So all of these caterpillars were parasitized, so we expect all of them to have parasitoid emergence if things are going well. And I'll orient you here to this plot. On the x-axis, I'm going to have my shock stage or heat wave treatments. And on the y-axis, I'm going to have the proportion of hosts within each treatment that had wasp emergence or didn't. So here's my control treatment. As we can expect, basically all of the hosts had wasp emergence. We have 1 or 2 that didn't, which sometimes just happens due to a failure in oviposition or parasitoid establishment. So for my heat wave treatments, we can see something extremely different is going on in the early treatment, where almost all of the caterpillars did not have wasp emergence. The middle and late heat wave treatments look pretty comparable to the controls. So what's going on with these early bugs? Are they pupating? Are they just dying? What's going on? We actually found a really odd result where the caterpillars in this treatment had extremely abnormal physiology and development. I'll show you a picture here. So on the left, we have a normal unparasitized Manduca sexta that is wandering, which is the stage prior to pupation. And on the right, we have one of my without wasp emergence hosts, or WOWIes. So we saw that these caterpillars lived an extraordinarily long time. A normal caterpillar will wander between 12 and 16 days after hatching. These caterpillars were living up to 35 to 50 days after hatching, which is just unbelievably long for this species. If you can also see in this picture, this WOWIe is much larger than the unparasitized wanderer. And we saw that these caterpillars continued to eat throughout the majority of their lifespan and gained up to twice as much mass as a normal wanderer. So this wanderer is about 10 grams. This WOWIe is about 20 grams. And even more interestingly, a normal wanderer will pupate and go on to metamorphose into an adult, but none of these WOWIes did. They either died as caterpillars, a few attempted to wander and died, 1 or 2 attempted to pupate and failed as a larval pupal intermediate. So something really odd is going on in this early heat wave treatment. So now let's talk about what happened to the parasitoids. Again, on the x-axis of this plot, I'm going to have my heat wave treatment. And on the y-axis, I'm going to have the average number of parasitoids per host. And the colors within these bars indicate the final stage achieved by these parasitoids during the experiment. So if we look at our control, we can see that about 50% of this bar is yellow, meaning that about 50% of parasitoids survived through adult eclosion and would be able to go on to reproduce and create the next generation. You can see there's also a portion that emerged but failed to eclose. They either failed to spin cocoons or failed to complete pupation. And then there's another portion that were mature second instars that remained within the host. And I discovered and counted these by dissecting all of the hosts after they exited the experiment. And this is pretty typical. Most, in good conditions, parasitoids of this species, Ctesia congregata, will have about 50% survival to adulthood. So then let's look at my heat wave treatments. Unsurprisingly, we have a very, very small bar for my early treatment. These were for the 1 or 2 caterpillars that did have wasp emergence. I dissected them. They had very, very few wasp larvae within them. So looking at my middle and late heat wave treatments, we can see the middle is pretty much comparable to the control, but something very different is going on with the late heat wave treatment. And I'll remind you, reminding you of my previous plot, all of the hosts for this treatment had emergence, but we can see that they had very low numbers emerging and very low numbers surviving to eclosion. That yellow bar up there is extremely small, and that's about 2% of parasitoid loads actually surviving to adult emergence or adult eclosion. We can see that the vast majority of this treatment, parasitoids actually got stuck in the host and perished as a mature second instar or an immature second instar. So this gives us an idea that something is going on here. These heat waves during the late stage of development is causing really high mortality and is also causing developmental asynchrony, where normally these parasitoids develop at roughly the same rates that they're all ready to emerge at the same time, as it happens in a very short window. So my results found that, yes, Ctesia congregata has different temperature sensitive stages in parasitoid development. They have different thermal tolerances depending on their developmental stage. And we saw that they had 2 temperature sensitive stages, one early and one late. I'm going to briefly go through some possible hypotheses about why. We have some really cool work going on in the lab right now that's hoping to disentangle which of these is more physiologically probable. So early on in parasitoid development, I told you that there's this polydnavirus that has a really important role in suppressing the host immune system. When you don't have that virus functioning normally, you can see things like this in this image, where you get lots and lots of black dots in the host tissue, indicating that these eggs have been, these are parasitoid eggs that have been encapsulated, melanized, and killed by the host's immune system. So it's possible that these early heat waves are disrupting the polydnavirus, killing the parasitoid eggs, but there's potentially enough polydnavirus still around, or the function of the polydnavirus recovers enough to disrupt the host's hormonal system so that they cannot pupate and wander, like I showed previously. Alternatively, it could be that the parasitoid eggs are just very sensitive to temperature. The polydnavirus is not being affected, and the parasitoids are being killed by the heat before they can hatch or complete their embryonic development. So what might be happening in the late heat wave, where we saw this really high mortality and this developmental asynchrony? Well, the parasitoid's second instar, when they experience this heat wave, is really important for nutritional uptake. This is when they put up on their vast amount of their mass. They increase in size about 35 times, and it's really important for them to get this nutrition in order to emerge. There have been a couple of studies that have shown that if you starve the host during the parasitoid's second instar, you get increased mortality the longer that host is starved. However, it's also a really important time for hormonal regulation for the parasitoid. They need specific hormones at specific levels in order to emerge successfully. So the heat wave could either be disrupting nutritional uptake or the regulation of these important hormones and causing this developmental asynchrony and this inability of the parasitoid to successfully emerge from the host. So to sort of broaden back out, we've seen that there is stage-dependent thermal tolerance, at least for this parasitoid, Catesia congregata. The reason why I was interested in looking at this is, again, we have this increasing frequency of extreme temperature events due to climate change, and that organismal and ecological outcomes, as we've seen in my work, can be really dependent on the developmental timing of these heat waves that are that they're experiencing. So it's interesting and important to study how these extreme temperature events are going to impact organisms based on developmental timing. I'll also remind you that this is a multi-species system. We've pretty much focused on the parasitoid, but host tolerance is also dependent on developmental stage, and that could play into this relationship as well. And like I said in the beginning, parasitoids are an important top-down control, both in agricultural and natural ecosystems.
4 minutes.
Thank you. So if we're seeing higher parasitoid mortality, we could see cascading effects on the host population and on the ecosystems in which they both live. With that, I'll get to my acknowledgements. Big thank you to everyone in the Kingsolver and Willett Lab that have helped me with this work. Thank you to my committee, to the NSF for funding, and a really big thank you to the organizers and moderators of this conference. This is my first time, and I've had a really great time so far. And I'm happy to take any questions. And my email is there if people want to get in contact with me for things I didn't get to.
Great, thanks Elizabeth for a great talk. So people should feel free to send questions in the Zoom window or through the Slack channel. And I had a question. I was curious, do the caterpillars take advantage of these susceptibility windows if they're in a natural setting to like thermal regulate to disrupt the parasitoid? Is there any evidence of that?
Yeah, we've looked into that a little bit in my lab. Manduca sexta is a really big thermal generalist, so it can survive and do very well at a broad range of temperatures. So it really doesn't have a whole lot of thermoregulatory behavior. But we have done some pretty cool studies. People in the Willett Lab have looked at where caterpillars choose to be on a hot plate, depending on if they're parasitized or not. And I did a field experiment looking at location on tobacco plants, and that doesn't really seem to change between parasitized and non-parasitized. It could happen before parasitization, but we haven't looked at that yet.
Very cool. Okay, we have some questions from Mike. Do you expect the parasitoid to be able to adapt to increasing temperatures or heat waves?
That's a really good question. And I think it really depends on what part of the physiology is being affected in these temperature sensitive windows. So if it is something that they can easily adjust, like if they are sensitive in the egg stage and they can develop, better heat shock proteins or, things like that, that could allow them to. But I don't actually have a really good sense. But I think we'll know more once we know what physiological sort of pathways are being affected.
Awesome, thanks. And just as a correction, that was from Jimmy, not Mike. Okay, there's one more question, but I will post it to everyone. And Elizabeth, if you can answer that in the chat, that'd be great.
Okay, cool. That was a great talk, Elizabeth. Thank you. So our next up is Volker Rudolf. Volker will be presenting a talk entitled Linking Phenological Synchrony to Population, Demography, and Species Interactions. Take it away. Volker, you are muted.
I was so excited about sharing the talk that I forgot about unmuting myself.
Great, we can hear you now.
All right. All right, I want to take this chance to talk about the effect of phenological synchrony on population demography and species interaction. And this is work I've done with collaboration with Shen and Carter. And so most of us are probably right now painfully aware of the fact that seasons can change quite dramatically when we look at abiotic conditions, right? Can be very cold or very hot, dry or wet. And as the year progresses, we see these dramatic changes. And it's probably not surprising that because of these changes, the majority of organisms have go through changes in these key life history events. And these key life history events occur at specific seasonal time periods. If you think about flowering, migration, colonization, reproduction, and so on, all of those things occur at specific times of the year. And phenology have received a lot of attention in the past decades because they're one of the best documented responses to climate change. In fact, if you look across systems and across species and taxa, we see a global effect of climate change on the phenology of species. Also, not everybody is responding the same way, so we see a shift in interactions as well. Now, one of the big questions here is been to understand how phenologies in general influence species interactions and how we can predict the consequences for climate change. Now, we traditionally taken out calendar-based view. We ask, when do these events occur, right? So we focus on things like the first flowering or the first reproductive event or calling, or when do species come back from their migration. The problem with this is that at the population level, we really don't just have a single event, but we have individuals and they don't always do the same thing at the same time. So really we have some sort of distribution of phenologies, and this distribution can either be very tight when everybody's very synchronized, or we can have examples where phenologies are really far spread out. We have a low level of synchrony, and we can have, of course, anything in between. And what's interesting about this is that we see this difference in phenological distributions across species, but we can also see those within species, and there's good evidence that these distributions are changing in response to climate change as well. So why worry about those specific distributions? Now, you can think about what's happening inside a population and how that's linked to the synchrony of populations. So at high synchrony, we basically have a species that is present for a much shorter period of time in a given phenological stage than if you have low synchrony, right? So the duration and the timing are affected by this. Within population, however, we can also think about what do individuals experience. And with high synchrony, everybody sort of experience the same kind of like type of conditions. Whereas with low synchrony, you really see kind of like individuals that may experience different conditions, either in abiotic conditions or species interactions. So we get more variation within population. Of course, as everybody is highly synchronized, we also get higher densities in contrast to low synchrony, whereas like populations are sort of more spread out over time, so you have an average lower densities in the population. Should also point out that in this scenario, of course, we sort of have an example where individuals can also experience different densities depending on their respective phenology. Finally, if you sort of think about populations that sort of are consisting of things like offspring, like after germination or reproduction, we of course also can see differences in individual traits, because individuals at any given point of time will be a mix of old and like young individuals, small and large, and we get some sort of trait variation at the population level. So one of the questions we've been asking in the past few years was, how does this phenological synchrony really affect the demography of populations and species interactions? What are the underlying mechanisms? And are there conditions when those become more or less important, right? Or can we go back and sort of just keep track of our single metrics to answer these questions? So we've done a number of experiments, but we sort of want to get a general understanding across different conditions and decided to take an interval-based modeling approach that sort of represents a basic life cycle of like a seasonal species. And just to walk briefly through that model, essentially what we have is individuals are initially dormant or like absent in the environment, they're not active. And then once the phenology basically tells them they become active and so start interacting with members in the community and consuming resource. What we can do in the model, however, is we can manipulate the synchrony of these individuals in the population. We can either have them all show up very synchronized at a given mean, or we can increase the variance around that mean. And for now, we're just going to focus more or less on normal distributions, but obviously this can be manipulated based on your personal preference. All right, so once individuals become active in the environments, we can also they consume resources based on some basic function of resource densities and the size and development stage, and they then convert those resources in growth. We do a couple of checks and balances. We're making sure that individuals actually eat enough to meet the basic metabolic and bioenergetic demands. If they don't, they starve. If not, they can continue to grow. And this is kind of like how competition enters the system as well. Then once we continue to grow, they have to decide whether it's time to move on to the next developmental and phenological stage. So once they reach a minimum size threshold, they then use an optimality approach to decide whether the conditions weren't to stay in the environment or whether it's time to move on. And this could be metamorphosis, this could be like dormancy, it could be reproduction. Really, it depends on the system. And what I wanted to do here is kind of like emphasize that this is not a model specific for tadpoles. While that has inspired it because we work in the system, it could be just as well applied for anything else, including like plants or insect or whatever you're thinking about. And we try to keep it really simple and really just focus on the main dynamics. Now, what is exciting about this model for us is that we can do things that we weren't able to do in an empirical system, and that is to starting to separate the different mechanisms that I talked about and how they're linked to the dynamics of the system. So in this scenario, what we see here is we see that there is variation in size. And we know that variation in size is typically linked to things like resource consumption, right? So here you see that small individuals shown on the x-axis from left to the right start consuming more resources if they increase in size. And at the per capita level, when they both occur, that generates asymmetries in their per capita competitive effects. And we refer to this as competitive asymmetry. And that could be one of the key drivers of phenological shifts and phenological synchrony, but we really wanted to test that. So in our model, we can manipulate that strength, and we can even completely remove it to a point where the difference in timing is disconnected from your competitive ability in the system. All right, so let's jump in and take a look at some key demographic rates of populations. So I'm showing you here these 4 panels are key demographic rates like survival, per capita mass, biomass export at the population level, and so when individuals move to the next stage. And on the x-axis, you see high synchrony to low synchrony, which could vary across a gradient in the model. And then on the y-axis, you see kind of how this change in synchrony altered that respective response variable. So we do this in comparison to a highly synchronized population, so I guess a reference. All right, so the key take home from this is really that synchrony affected all measured demographic rates in the population. And to really sum that up, essentially what you get if you decrease synchrony, you have reduced survival, but the individuals that survive are larger and they advance to the next stage faster. This is exactly what we've seen in our experiment, suggesting that we really have a nice representation of the natural dynamics and synchrony effects in our models. Now, when you look at the different colors that represent the different levels of competitive asymmetry, in the gray circles represent the scenario where we had no competitive asymmetry. So in other words, it didn't matter when you hatched, it had no consequence for your competitive ability. And you see that in this scenario, you still get effects of phenological synchrony. So the idea that the trait differences that drive in the dynamics are not important necessarily, it can arise simply because individuals experience different environments because of this difference in phenologies. This said, competitive asymmetry strongly enhances the effect of phenological synchrony, and we see some somewhat non-linearities that we can also detect in some of the empirical systems. Now, because we can follow individuals in the model, including those that died, we can also look at some fitness metrics and look at selection in these different scenarios. So that's kind of what's shown here on the y-axis. Then the x-axis, we look at how that fitness relates to the relative hatching date of individuals within a given population. And if you're on the left, you hatched early on average than the rest. If you're on the right, you hatched later than most individuals in the population. And so regardless of the level of competitive asymmetry, we see the same happen even at high synchronies, that early hatching has always been advantageous. So individuals always have a higher fitness than late hatching, which is not particularly surprising. Competitive asymmetry enhances that selection pressure, and that's also what we expect. And low synchrony really strengthens that election. So we get a stronger level of selection, stronger fitness differences in populations that are at low level of synchrony. So at the population level, we have a good understanding of the mechanism in the individuals and what's happening, but we wanted to really know what happens with species interactions. Because one of the big things that people have reported and we've seen is that climate change has altered the relative timing of interactions. And we wanted to know how does that interact with the phenological synchrony within population. So we took our model, added a competitor species, and then we allowed individuals to either arrive on average before their competitor, at the same time, or after. So we can look at the results here, and what we're seeing here, again, is the same setup where we have synchrony on the x-axis, and then we have a response variable on the y-axis. And I want you to focus first on the top panel, that's proportional survival. So the circles, the green circles, show that example where individuals hatched on average before the competitor, and we see the same pattern that we saw before, that is low synchrony, reduced survival. But if the individuals hatched on average after the competitor, that was completely reversed. In other words, the timing of competition could reverse the effects of phenological synchrony within population. So we get a context-dependent effect, and because of that context-dependent effect, we also see something else that's quite interesting if you think about the consequences of shift in interactions. And that is, if you sort of look at high level of synchrony, we see that the difference in arrival times are quite big. Whereas at low synchrony, the treatments are very similar to each other. And so that suggests that low synchrony in population can actually buffer against phenological shifts in species interactions. We can also look at the selection in the system again and looking at relative fitness. Here the setup is the same as I showed you before, except that the panels represent the different timing of the competitor. And what you're seeing here is that, again, early individuals do generally better, but if the population average hatches after the competitor, that selection is almost flat. There's almost no advantage, and that's because everybody does terrible if there's high synchrony. But if you then allow for low synchrony levels, to see that the lower the synchrony, the stronger the selection becomes in the system, the stronger the fitness difference between the early and the late hatching individual. So we get an interactions between the timing and the synchrony that determines fitness difference in selection in these populations. So why do we see this context-dependent effect of synchrony? Well, we can look at this scenario where the blue population arrives after the red population in this scenario. At high synchrony, everybody does poorly. At low synchrony, you got the lucky few that actually manage to hatch early on in the system. And they start to escape the competition, and they're the ones that really driving the demography of the populations in the system. So what I'm showing you here was this idea that phenological synchrony provides a lot of important information because it influences a number of factors at the population level, such as the timing, duration, and density, that all scale up to determine what happens at higher levels, including population demography,
4 minutes,
dynamics, thank you, interactions with other species, phenological shifts, and ultimately the response to climate change. So the argument here is that we really need to start paying a little bit more attention about phenological synchrony if we want to understand natural population dynamics and species interactions and the consequences to climate change. And my call here would be that we know surprisingly little about distribution of phenology, so a lot more work empirically needs to be done to look at this as well. So with that, I would like to thank everyone for listening. I want to again put a big shout out to Chin and Carter, who's done all the hard lifting from the modeling component and the analysis, and thank you for listening.
Great, thanks Volker for a really interesting talk. Anyone who has questions for Volker, just send them my way in the Zoom window or on the Slack channel. We have about 3 minutes for questions.
And anybody has questions, you can always email me later on, send me a message in Slack or something.
Okay, so we have a question for you. How did modeling the resources work?
Yeah, we agonized a lot about this. Essentially what we did a seasonal resource that's shows up the environment at a certain time. And then we had a non-renewable consumption of that resource that's based on the individuals in the population. And what that does, it prevents individuals that arrive too early from doing well because there's no resource, so there's some limit to how early you can show up. And it was more inspired by what we see in our temporary ponds. There are a number of other ways of modeling this. We looked into this. but we did a number of different sensitivity analyses that show that the pattern I showed you here is like surprisingly robust to like any variation in many of those parameters. So at least we feel pretty confident that it wouldn't really change too much overall. But yeah, we spent a lot of time thinking about that.
Do we have time for one more question, Sasha? Or
Yeah, I think so.
okay, would you expect high synchrony host populations to create greater vertical transmission bottlenecks on their parasites than low synchrony host populations?
Can you repeat it again?
Would you expect high synchrony host populations to create greater vertical transmission bottlenecks, or greater vertical transmission bottlenecks on their parasites than low synchrony host populations?
Greater bottlenecks. I'm not sure I fully understand the question in here.
Okay, I'll send it to you in the Zoom chat, and then you can think about it a bit more and respond.
Happy to chat more about it.
Cool. Thanks for that really cool talk, Volker. Our last talk of this first half of the session is coming up, and then we're going to hear from Regina May Francia, who's going to be giving a talk entitled Microclimate Amelioration and a Cross-System Comparison.
Hi everyone, thank you for being here today. My name is Regina May Francia, and I'm currently a master's student at California State University, Los Angeles. I'm very excited to be sharing my project, which looks at an across-system comparison of microclimate amelioration. So according to NOAA's 2019 Global Climate Summary, by 2100, the planet's average temperature may increase by 1.1 to 5.4 degrees Celsius. And we can already see this temperature increase when looking at recent temperature trends. Here I have a figure of the temperature trends from 1990 to 2019, where the red areas represent regions that have already warmed up to 1 degree Fahrenheit, and the areas in blue have cooled up to 1 degree Fahrenheit. And so with these temperature trends and future implications of climate change, different ecosystems may be affected by these changes in macroclimate. And so the presence of vegetation becomes even more important because vegetation has the buffering capacity to mitigate temperature extremes. And as an example of this is a figure from a study by Dufresne et al. 2019, which looked at 3 different ecosystems: deciduous, evergreen, and mixed tree forests. In that study, they found that temperatures in the understory of these forests were reduced compared to the temperature outside of the forest, thus showing how vegetation can buffer temperature. Now, looking across different ecosystem types, we infer that vegetation types may differ in their ability to buffer macroclimate, and this can be due to the stature of species, ecosystem conditions, or other abiotic extremes. Now, this mechanism is called microclimate amelioration, and microclimate amelioration is the process in which plants can mitigate harsh abiotic conditions. In the last slide, I talked about temperature, but this is also relevant for other abiotic factors such as wind and solar irradiance. Now, just to clarify the differences between macroclimate and microclimate. So to our left, we have this larger bubble, which we are defining as the macroclimate. And this is a climate over a large geographical region. When we zoom in and look at the microclimate, we see that it's the climate of the immediate surrounding of the plant. And so we can measure the impact of vegetation on microclimate via these measurements of soil moisture, temperature, relative humidity, solar radiation, etc. Now, macroclimate data have been collected for the past century, and initially it's been hard to measure small-scale fluctuations in microclimate. But innovation has made it cheap and accessible in the last 10 to 20 years, so now we can collect data on both of these scales and measure how much vegetation is ameliorating abiotic conditions like temperature. For our study, we are interested in whether vegetation mediates temperature and whether it is good or bad for plants living under the canopies of their neighbors. I have posed a question here. When does temperature amelioration matter? Well, in the 90s, Burtness and Callaway proposed the stress gradient hypothesis. This hypothesis states that competition is more dominant in more mildly stressful environments, and as there is an increase in abiotic stress, we see that there is also an increase in the strength of facilitative interactions. And so strong facilitation can occur when neighboring plants ameliorate harsh abiotic conditions via microclimate amelioration. Facilitation actually can drive enhanced performance of the neighboring individuals by improving their abiotic and biotic environment. And we can see this here on the left, where the growth of this smaller individual is facilitated by this nurse plant. Thus, positive outcomes of facilitation can include increased survival, increased growth, improved water status, etc. Now, globally, we can see the effect of stress in alpine environments. Here's a study by Callaway et al. 2002, where they measured the strength of competition and facilitation in 22 experimental sites in these alpine, subalpine plant communities. From this study, they found that at high elevation, where abiotic conditions are more stressful, facilitation was the more dominant interaction among plants. Along with this, they saw that there was also an increase in survival at high elevations where the species was surrounded by neighboring individuals. In the study, we can see that there are more stressful sites that did lead to more positive interactions in a global study. This particular study examined alpine environments as a stress gradient, but theoretically, this principle should hold true for other stress gradients as well. So as we see global changes in temperature, we want to understand the degree at which different ecosystems can mitigate abiotic stress and how this translates to changes in the plant communities of these ecosystems. And while we know forests can cool their microclimate, based on past research, we need to make sure we understand how other ecosystems respond as well. And so microclimate measurements become an important factor to understanding how these ecosystems will change as a response to a warming climate. Okay, so going into this meta-analysis, we pose these questions. 1, do vegetation types have different effects on the degree of temperature amelioration? Do environmental gradients such as mean annual temperature and mean annual precipitation drive changes in temperature amelioration? And are there interactions between vegetation types, mean annual temperature, and mean annual precipitation? So, for example, do forests ameliorate temperature better than grasslands? For our literature search, we use Web of Science to extract articles from the last 20 years with the following key terms: temperature, humidity or VPD, and microclimate, microhabitat or microsite, and vegetation or plant, and facilitation or competition. These papers were accessed on August 26, 2020. From our first extraction, we had 305 articles, but from those, 62 of those articles included both microclimate metrics of either temperature or humidity, along with plant performance metrics. And for each paper, averages of these measurements were retrieved and a standard error was calculated. For this presentation, we specifically looked at air and soil temperature. We also standardized ecosystem types to include forest/woodland, shrubland/desert, and grasslands. We also extracted mean annual temperature and mean annual precipitation for each study site from the WorldClim database. For data analysis, vegetation effects were compared in each study using a log response ratio. We use this equation of the natural log of undervegetation divided by open. Also, multiple study comparisons were averaged per study, so we can get a single log ratio per study. We also calculated variance using scalc function in R and used the metaphor package in R so that each log response ratio was weighted by variance. We also analyzed the main effects of ecosystem type, mean annual temperature, mean annual precipitation, and all two-way interactions. And just as a note, grasslands were excluded from the results presented here because there were only 3 studies with temperature data. Now, moving on to our results. Here's a map with all the locations of all the studies we extracted, and the key here shows the colors corresponding to the ecosystem types. From these 62 studies, 24 of them were from shrubland/deserts, another 24 from forest/woodland, 7 from grasslands, and then 7 from other. Now, when conducting a 2-3-way interaction, we found significant effects of mean annual temperature on temperature amelioration. We also found an interaction between mean annual temperature and mean annual precipitation. And lastly, we saw a marginal significant interaction between precipitation and ecosystem type. For our first figure, we compared mean annual temperature and temperature amelioration for all studies. The dashed line here at 0 signifies that there was no effect of vegetation cover on the temperature of microclimate, while these positive values represent vegetation warming the microclimate, and these negative values represent vegetation cooling the microclimate. And so we found that vegetation generally cools the microclimate and that the degree of cooling is stronger at these hotter sites. With this next figure, we compared dry versus wet sites, with the mean annual temperature on the x-axis and temperature amelioration on the y-axis. Here we see that at drier sites, vegetation consistently cooled the microclimate on average, while at these wetter sites, the vegetation had the capacity to warm the microclimate in these cold ecosystems and cool the microclimate in the warm ecosystems. Now, the precipitation and temperature interaction may be due to the differences in vegetation type. Here we compared ecosystem type, shrubland/deserts versus forest/woodland. And we found that forests seem to cool more than shrublands, and that forests may also warm the microclimate in cooler areas. From our preliminary results, we found that the degree of vegetation cooling is stronger at hotter sites. At our drier sites, vegetation consistently cooled the microclimate on average. And that in wetter sites, vegetation had the capacity to warm the microclimate in cold ecosystems and cool the microclimate in warm ecosystems. Also, we saw that forests seem to cool more than shrublands and may also warm the microclimate in cooler areas. For our future directions, we would like to explore other microclimate metrics along with how it may affect plant productivity. Some questions we posed is, how is relative humidity affected by abiotic and biotic factors? How does microclimate amelioration affect plant performance on average? And then also, are there more positive consequences of microclimate amelioration in more stressful environments? That is the end of my talk. I'd like to thank my advisor, Dr. Alexandra Wright, our collaborator, Dr. Alessandro Filozoa, my funding from Cal State RISE MS to PhD program, and the Wright Lab. I will take any questions if you guys have any. Thank you.
Great, thanks, Regina. So for anyone who has questions for Regina, please send them to me in the Zoom chat window or on Slack. I think sometimes the time it takes people to write out questions slows them down, but if it ends up that questions come in, I'll send them your way, but we still have a few minutes to take questions. So if anyone's madly typing, we'll wait, don't worry. Great, a couple questions just came in. So the first one is, how much do you think these kinds of feedback loops will change the effects of climate change on ecosystem health?
That's a really good question. I think that hopefully this type of research can give us more answers to that question. And when we were doing like this meta-analysis, we did come across kind of two different types of questions. So my project focuses on the effect of vegetation on microclimate, but also there's that feedback that they're talking about where the vegetation can affect the macroclimate. And I think one of the papers I that I referenced, Defrene et al. 2019 talked about that a lot. And so hopefully the direction of this type of research is to quantify that type of effect that vegetation has on the microclimate.
Awesome. We have another question in the chat. Thanks for a great talk. This is from Joey, saying, I have a question more generally about the climate stress hypothesis you presented at the beginning. How is stress in that context defined? Is it an ecosystem level property or from the perspective of the individuals?
Yeah, I think, so with that stress gradient hypothesis, it definitely is on an ecosystem level. So looking at different papers, we saw that with alpine ecosystems, the stress gradient there was altitude. And then looking at different papers for this meta-analysis, they defined it as either it could be, for example, light in a forest setting, or it could be warming or cooling of a temperature in this larger setting. So I think definitely probably the ecosystem is what that stress gradient hypothesis is referring to.
And there's a follow-up to that question. I guess I was thinking, what happens if a particular something is stressful to one species but not another?
Oh, sorry, could you repeat that one?
Yeah, so I guess I was thinking, what happens, it says if a particular, there's a word missing, is a particular aspect or something is stressful in, okay, environment is stressful to one species but not the other. Sorry.
Yeah. So sometimes that is the case where in these studies where they measure microclimate, they're looking at a specific like target species. And so, yeah, the effect of stress is not, doesn't always have to encompass all of the species in that ecosystem. So definitely, I think it could be species specific when looking at how stress affects specific species.
Great. So one more question. Why do you think there are less studies in grasslands?
Yeah, so based off of my results, we did like retrieve less studies. Like I think there was like 3 with temperature data, but I don't think that is representative of all of the studies in the last 20 years, because for this specific meta-analysis, we looked at studies that had both microclimate metrics and plant performance. So that number might be a little bit higher if we changed our like meta-analysis search. But if there was a difference in how many studies focus on ecosystem, like specifically grassland, I definitely think that there might be this bias that the stature or the stature or the height of a species is a better ameliorator compared to grasses, which when they're among each other, they might be the same size. And so there could be some bias towards that, which is why it might be leaning towards like these larger like shrubland or forest ecosystems.
Great, thanks. And I think that brings us to the end. So
yeah, so that's the end of our first half. Maybe Mike can give any other instructions. All I know is that we now have 20 minutes break, and we'll come back at 20 minutes from now. So on the West Coast, 5:40, on the East Coast, sorry, on the West Coast, 2:40, on the East Coast, 5:40, and we'll get started right away with another great talk. So see you all in 20 minutes. Great. Okay, so our next speaker is Hengxing Tu or To, and they'll be presenting a talk entitled Priority Effects and Season Length Shape Long-Term Competition. Take it away.
Hello, everyone. My name is Hengxing Tu. I'm a second-year PhD student at Rice University. And today I'm going to talk about how priority effects and season length shape long-term competition dynamics. So we know that in nature, species arrive in their habitats at a certain time. And with some observation, we can easily tell that not all species arrive at the same time. For example, here's a graph showing different peak breeding activities of various frog species. And this is also true for the majority of interacting species, that they arrive in their habitats at different times, and their phenological difference would determine their temporal overlap, and therefore the extent of their interactions. And this kind of phenological difference has been one of the major focus of ecologists in the past decade, because as climate changes, different species will respond differently, creating more opportunities of phenological difference between interacting species and potentially change their interactions. While the majority of past studies has been focusing on the phenological difference between trophic interactions, the phenological difference between competing species, such as these different grasses on the right, is equally important. Recent studies indicate that the relative timing of arrival when colonizing a new habitat may cause priority effects between competing species and subsequently affect community sampling process. What makes the story more complicated is that most habitats are also seasonal, meaning that at the beginning of each season, there is a colonization process and therefore a potential for phenological difference between interacting species. And we also know that phenology could vary over seasons. For example, here's the relative abundance curve of common wood-nymph butterfly over time. And we can clearly see that the shape of this curve changes according to different climatic conditions of each year. So if the phenology of both competing species vary over time, their phenological difference is also likely going to vary over time. So apart from the exact phenological difference between competing species, this kind of seasonal variation may also contribute to the long-term competition outcome. So the question then goes, how does seasonal variation in arrival time affect competition outcome in the long term? To answer that question, we need to examine one consequence of such phenological difference, trait-mediated priority effects, which happens quite literally when the priority effects between the two species is mediated by the change of interacting traits due to its arrival time difference. For example, suppose we have two competing species, both stage structured and have very similar developmental rates. If the two species arise simultaneously, they are interacting with the other species in their own stage, and that will lead to interspecific competition very similar in magnitude. And this relative symmetry will also be maintained for all their life stages. But if one species arrives earlier, the later arriver will experience competition from early arriver in a larger size or stage than itself. And this will cause a competitive asymmetry because the smaller individual will likely experience larger competition from the larger individual. And this competitive asymmetry will also be carried over for all their life stages, likely change the overall competition dynamics of the two species. And in this case, the ontogenetic stage or size is a trait that changes according to phenological difference between the two species. So one way to quantify this kind of trait-mediated prior effects is by a competition phenology curve, which maps competition coefficients to the relative arrival time or phenological difference between the two species. And we can call it delta P for short. If the two species arrive simultaneously, delta P equal to 0. And in this case, they will share the same interspecific competition coefficients. But if one species arrives earlier than the other, delta P will not equal to 0. And we can clearly see a difference of the two competition coefficients in that the early arriver would have a larger effect on the later arriver. Now we have an idea of how phenological difference might affect competition outcome, but there is yet another factor at play, and that is life history. Seasonal systems have a diverse range of life history, from annual plants that reproduce once per season to zooplanktons that reproduce multiple times in a season. And this gradient of number of generations per season will also likely to contribute to the long-term competition dynamics. To understand why life history matters, we can consider two competing species with only one generation per season. And you can think of something like annual plants or pond amphibians. And their arrival time differ by delta P. So here, circle and square are different species, and sizes of these shapes reflect those of the individuals of the respective species. If we sample the community within the season, we'll find only one size of one species interacting with only one size of the other species. And this fixed size difference is determined by delta P. So in this case, the effect of delta P on competition actually persists throughout the season. But if these two species have more generations per season, such as in the case of zooplanktons, where there's a continuous reproduction throughout the season, both species will start to reproduce in a second generation. And each species will not only interact with one single stage of the other, as in the community composition on the left, where there's only one generation. Instead, both species will simultaneously interact with size distribution of the other species, as shown in the community composition on the right, starting from the second generation. And this will change the overall competition coefficients because overlapping generations will make the initial size difference less important, and therefore diminish the effect of initial delta P and reduces the importance of phenological shifts. So to fully capture all these complex dynamics, including phenological difference, its variation, and life history, we use a stage structure population model, in which we have 5 juvenile stages and 1 adult stage. Each juvenile stage can move into the next stage by a transition probability. Adults can survive for more than one time step and reproduce to form new cohorts of stage 1 juveniles. And this will allow for overlapping generations by continuous reproduction. And all these transition probabilities are scaled by both intra and inter-specific competition from individuals of all stages. And finally, at the end of each season, all individuals die except for a proportion of stage 1 juveniles produced by the final adult population. So in our model, the length of each generation is fixed, and that is 6 time steps corresponding to the 6 life stages. And we use season length to reflect numbers of generations per season, such that longer seasons will contain more generations. So we examined the competition results by looking at invasion growth rate and ran simulations with different parameters. And here's the results. We have number of generations per season on the y-axis and delta P on the x-axis. And the competition outcome of each parameter combination is represented by color shown on the right. So first, looking at 2 generations per season, we see that as phenological difference changes from -5 to 0 to 5, which spans from species 1 arrives early to simultaneous arrival to species 2 arrives early, the competition results change from species 1 wins to numeric priority effects to species 2 wins. And here numeric priority effects means that both species cannot invade the other species, so that the species with the larger initial population will win. Now we look at results along y-axis, and we see that the competition outcome also changes with different numbers of generations per season. And importantly, when there are many generations per season, we see that the initial delta P doesn't matter at all, as the competition results don't change along the x-axis. We then added seasonal variation of delta P by using a random delta P at the beginning of each season. We ran 30 stochastic simulations per parameter pair in the black rectangle, evaluated results by comparing the endpoint populations of the two species, then plotted the proportion of each outcome out of all 30 simulations by a pie chart. And here's what we found. And again, x-axis is phenological differences, and y-axis is number of generations per season. And as we expect, we see variations of competition outcome for each parameter pair, as shown by these different colors in each pie chart. And we still see changes of competition outcome along the phenological differences, or along the x-axis. As we look at different number of generations, we see that with more generations, the effect of both phenological difference and its seasonal variation diminishes, such that at 20 generations per season, there's no alternative competition results for each parameter combination, as shown by these single color pie charts, and no changes along the x-axis as well. So what are the underlying mechanisms of our observations? So far, we have been assuming an underlying competition phenology relationship that looks like this, which describes the trait-mediated priority effects. But most previous priority effect literature assume that the competition coefficients do not change over the LP. So we then did simulations under this assumption and compared the results. On the left is the result with trait-mediated priority effects, as we see earlier. And on the right is where we assume no relationship between competition and phenology. So without this relationship, the majority of parameter space show numeric priority effects, meaning that the more abundant species wins regardless of phenological difference, except for a few extreme cases in which one species wins. We also don't see an influence number of generations per season on competition results with most parameters. Adding seasonal variation of delta P, and again on the left we have trait-mediated priority effects, and on the right with no such effects, we also don't observe alternative competition results for the majority of parameter space, where you can see that there's only one color for most pie charts, which is fundamentally different from what we observed on the left with the competition phenology relationship, in which we do see alternative competition results as shown by these pie charts with different colors. And we also see that without trait-mediated priority effects on the right, the competition outcome also doesn't change along either x or y axis for a majority of parameter space examined, which is different from what we observe on the left. Now we turn our attention to life history. In previous examples, we have shown that for species with only 1 generations per season, the effect of delta P will likely persist, while with more generations per season, the overlapping generations will diminish the effect of initial delta P. So we hypothesize that overlapping generations is also an important factor in determining competition outcomes that we observe. And this is exactly what we found. On the left is the base model with the overlapping generations as we have previously seen, and on the right is the result when we take away overlapping generations by disabling continuous reproduction in the model. And we can see that although phenological difference still affect competition outcome, as we can see that there's a color change along the x-axis, number of generations per season on the y-axis doesn't change this result at all. So this means that overlapping generations is also an important contributing factor to the observed dynamics on the left. And finally, we look at the realized interaction strength over a time period for both species, weighted by their stage distribution, and this is with a specific fixed phenological difference. And we see that the competition from and experienced by each species changes dramatically over a number of generations per season. And that indicates that the changing number of generations per season alters the relative competition strength of the two species, and therefore the competition outcome. So to wrap up, if we consider a community as a point in this plane where the two axes are the two interspecific competition coefficients, and we start with point A, where both interspecific competitions are the same. When we add delta P, where it's seasonal variation, that will likely shift this point toward regions with alternative competition outcomes, such as B or C, or anywhere in between. But extending the season counteracts this effect and drags the community back to its original point by diminishing the effect of delta P. And this is also true if the starting point is in another region, such as point D. And where exactly the starting point lies depends on the magnitude of intraspecific competition. So we see that the effect of phenological shift for a seasonal competitive system not only depend on how much the shift is, how variable it is among seasons, but also how many generations there are in a season. And this is especially interesting because if we think of the diverse seasonal systems in nature, their numbers of generations per season ranges from 1 to many, and that might affect how they respond to phenological shifts due to climate change. So one prediction is that fast generating systems such as zooplanktons might experience a smaller effect of phenological shifts because of all their overlapping generations, while slow generating systems such as annual plants might be more affected because their initial phenological difference is more persistent. And with that, I would like to thank my advisor and co-author, Volker Rudolf, as well as Josh Fowler, Tom Miller, and Joe Neal for their comments. And any questions?
Great, thanks so much. So for anyone who has questions, feel free to send them to me in the Zoom chat window or send them on the Slack channel. We have about 8 minutes for questions. Okay, so we have a question in the Slack, or sorry, in the Zoom window. When you run your models with multiple seasons, do you set the initial frequency in the next season by the final frequency of juveniles, or is the phenological differences reestablished in seasons 2, 3, etc.?
Yeah, so we set the initial population at the beginning of each season directly related to the adult population of the last season. So basically the adult reproduce, and then we kill a proportion of its offsprings, and then we carry that proportion of juveniles, stage 1 juvenile population into the next season. So that's how we set up the model. So basically we still got the rollover, so that the population at the end of the previous season is directly related to the population of the next season.
Awesome, thanks. Another question from the chat. I would imagine that size asymmetry also has a strong effect on facilitation. In particular, larger individuals can reduce temperatures and increase humidity for smaller neighbors. Did any of your models allow for an alpha value to reflect facilitation?
We have not included this. So far, the alpha values are strictly reflecting the competition between individuals in the same habitat, but we do see that it's possible that the individuals might facilitate each other, and it can be easily done by modifying the competition phenology curve that I just mentioned to include some kind of like, if we're talking about competition coefficients, then negative, but if we're talking about some other measurements of facilitation, then positive relationships of that kind of parameters, and that might change along the phenological shift. So I think that can be easily done, and I think that will be very interesting to look at.
Great. We have another question from the chat. How does the competitive effect of an individual change as it moves between life stages?
So what we assume here is that the size or life stages of a species or of an individual is directly affecting the competition effects that it can exert on other individuals of the same species or of the other species. So we assume that if we have a larger individual, that might cause a higher competition coefficients onto the other individuals of a smaller size of itself, which has been tested in the field empirically for many studies, systems. Although that might not be true for some other particular systems, but for now we're just assuming that it's the case in the model. Yeah.
Awesome, thanks. So we still have about 4 minutes for questions. If anyone has been madly typing, you can hit enter or start typing now. Okay, so we have a question through Slack. What was your modeling approach? Did you use an individual-based approach, stochastic recursions, or something else?
It is just matrix population model. So it's like the simplest form of that, except that it has like 2 species and 6 stages. So it's like a huge matrix, if you will, but it's still following the basics of the model of the matrix population model. So that, you know, like there's a transition matrix, and each of their entries are scaled by some kind of density dependent factors. And then we multiply this transition matrix to the current population vector, and that will result into the next generation of, next time set of the populations. And we do, as I said, take some different approach when we end each season. We assume that the beginning of each season, only juvenile stage 1 juveniles enter the system, and that is directly related to the adult population of the last season.
I forgot to read the very start of that question, which was awesome talk. So
thank you.
So if there aren't more questions right now, I actually didn't do a great job introducing the second half of our session. So maybe I can take a second and just say basically what I said at the beginning of the day. So I'm Sasha Wright, and I'm your moderator. Scott Taylor is here, and he's handling questions. When you have questions at the end of a talk, you can send them through multiple different channels. You can ask them here in the Zoom chat. You should be able to send a private message just to Scott Taylor. You can also send them by direct message in Slack to Scott, or in our climate session channel in Slack. I think that's the basic rules of the game, and maybe I'll go ahead and introduce the next speaker. I'm just looking at my notes to make sure yeah, okay. So our next speaker today is Collin Edwards, and Collin will be giving a presentation today entitled Shifts in Rare Butterfly Phenology Are Associated with Increasing Population Abundance. All right, take it away.
All right, you can hear me? Looks like yes. All right,
Yes.
Thanks for coming. My name's Collin Edwards. Today I'm going to be talking to you about some work that I've been doing with Elizabeth Krohn and with Cheryl Schultz. Oh, there we go. And they look like that. So one of the things that I would say one of the downsides of working in ecology is reading about or working on projects in which we find populations are going extinct or populations are declining. We've been seeing this really rapid increase in population declines and extinctions in the last few decades. That's driven in large part by anthropogenic forces, particularly climate change and other stuff like habitat loss and invasive species. But there are a lot of species, particularly insects, that are in trouble. We're also seeing pretty dramatic responses in phenology, sort of the timing of activities in response to climate change. So this is a plot showing the shift in the spring timing. So when different species are active at the beginning of their life cycle. And the negative values here are showing a lot of species that have shifted to be more advanced. So they are active earlier in the year. Now, these two things might be two separate signs that things are going poorly and a lot of species are in trouble. But they might actually be linked. So those species that are shifting their phenology to the earlier may actually be essentially tracking climate change to some extent, and that might be mitigating potential issues in terms of population growth. And in fact, recent studies have shown that that is the case, at least for some taxa. So I'm going to talk mostly about 2 recent butterfly papers because this talk is about butterflies. One of my lab mates, James McKlin, has this awesome paper that came out this last year in which he looked at 89 species of butterflies in Massachusetts and looked at how their phenology shifted and how their abundance shifted. And this is a plot of that start of activity on the x-axis. So negative values means that those populations have been shifting to be active sooner. And on the y-axis, we have their change in abundance. So essentially, is the population growing or shrinking? And what we see is that those populations that are shifting their activities earlier in the year are also the same populations that are increasing in abundance. A similar study, McGregor et al. From 2019 in the UK, with a different set of species, found that same general pattern. The x-axis has switched here. So now instead of the change in start of flight, we're looking at advancement. But we see the same pattern. So as the species, well, for the species that advance more, their populations are also generally growing. So that's the talk. Thanks for coming. No, there's a couple of caveats here. So the first is that these 2 studies are using common species, which makes sense because they needed enough data to estimate population trends and phenology. And in both cases, the patterns that we saw were driven mostly or entirely by multivolting species, so species with multiple generations in a year. And what it looks like, I'm most familiar with the Massachusetts butterfly data, but what it really looks like is that these multivolting species, the ones that shift their phenology earlier in the year, they're actually finding time to fit in 1 more generation at the end of the year. And that is probably why their populations are growing. So that is a pretty qualitatively different thing than simply tracking climate change. So we wanted to know, is phenological advancement good for populations of rare butterflies? We don't know if the trends hold for these rare butterflies, and also for univolting species, where they're not fitting in extra generations, but something else is happening. So the rare species in particular are kind of a problem. There are a lot of species where we don't have clean population estimates or estimates of the timing of their activities. That is most common for species that are rare. It's hard to get good data on them. So to tackle that, we brought together data from 21 species that are at risk. So we think they're in trouble where the data comes from. And they're all univolting, at least where we gathered the data. There are a few species on here that are multivolting in other parts of their ranges. So I'm fairly new to working with these species. I certainly don't expect you to recognize or remember all of the names, but I was pretty excited in reading through them to realize we have the makings of a pretty great superhero reboot, but with butterflies. We've got such charismatic names: Lang's Metalmark, which actually looks like this, Long Dash, who's actually about like that, and the Frosted Elephant, of course, who looks like this. So it's not related to the rest of the talk, but if anyone knows the Rousseau brothers, I've got some ideas. So in terms of the methods, I want to focus in on just one of these species to walk through what we're doing. And so I'm going to talk about the Lang's Metalmark because I really like the name. So this is a species, a subspecies rather, of the Mormon metalmark. And the caterpillars feed on plants in the buckwheat family. And in the day, they kind of roll up inside of leaves to hide from predators. And then at night, they come out to feed. This species is subspecies, is critically endangered. It is found in one place, as far as we know, which are the Antioch Dunes, which are just a little bit east of San Francisco. So we have quite a few years of data from this one population because scientists have been trying to study them and understand what's happening to them. Here's another picture of these beautiful little guys. So the kind of data that we have for butterflies and for a lot of other species are transect counts. So in this case, 10 times throughout this year of 2007, scientists went out and they walked the same route and they counted the number of butterflies they saw. And it looks like they saw butterflies on 5, maybe 6 of those times. Yeah, and so we have about 800 years of this kind of data spread across our different sites. What we really want is to take this collection of points and to estimate the actual population dynamics, so the population size and the phenology. But we want some way of estimating that's better than me just hand drawing in a little curve there. This seems like it should be a pretty simple problem. We have data points, we want to fit a statistical model to it. And in fact, a lot of methods have been proposed. there are a lot of really elegant approaches on this list, and some of them are good at fitting very different shaped curves. Some of them, like the Zonneveld model, are directly linking the shape of the curve with aspects of butterfly biology. So when you fit the data, you're inherently learning about the mortality rates or the lifespan. But there are a couple of problems with all of these methods here, at least for our data. And the first one is that they are generally developed with the idea that you have a lot of observations in a given year. For example, this is from Clark and Thompson 2011 with the generalized epsilon skew Gaussian, which I really like as a sort of a mathematical approach, but they're looking at 20 to 40 observations per year. This one, where we have 5 non-zero observations in the year, that's actually really good for our data. And so the methods that I've listed here, they simply don't work for our data. There is another challenge, which is maybe a little bit less relevant for us because I really like digging into statistical approaches, but most of the methods listed really require a pretty good understanding of statistical computing. So the Zonneveld model and the generalized additive model, those both have software implementations that make them pretty accessible. But the rest of that list, even though there's, 7 different methods that have been proposed, they've been cited and used in papers a total of 6 times. So even though we have this plethora of data or of approaches, it doesn't work for our data. And in fact, it seems like maybe that's not super accessible for a lot of ecologists. So the answer that Elizabeth and I have been working on is to fit our data with Gaussian curves, which are basically normal distributions, except they're not a distribution anymore. So they don't have to sum to 1. We have a manuscript in the works. The preprint is online. It kind of goes into the details. I want to get to the results, so I'm going to keep my explanation brief. But the general intuition is that a Gaussian curve is defined by this equation, which probably looks kind of intimidating, but the piece that we've exponentiated there is actually just a quadratic equation. And so that means if we take the log of both sides, we just have a quadratic function on the right. And that's something that we can fit really easily in R or any other statistical programming language. In this case, we can fit it with a generalized linear model where we just have 2 terms and we use a log-link function. The results I'm going to show you today are slightly more elaborate. We've been using a mixed model for reasons that I'd be happy to get into. So we have our approach. We can fit not just our 2007 data for our Lang's metal mark, but in fact, we can fit the vast majority of our data. And that gives us things that look like this. So here I'm plotting the most recent 10 years of data for Lang's metal mark. And we can immediately see that there are trends in the abundance and maybe also in phenology. It sure looks like as we move upwards, so more recent in time, the population sizes are getting much smaller. And maybe they're also shifting to be a little bit later. Now I could try to tell a story about this one population for this span of time, but we have a lot of populations. So we need some ways of summarizing this information. And I'm going to walk through 4 different metrics that we've been using. So the first one is pretty straightforward. We want to know the size of the population in any given year. So I'm going to focus in on 1998 to illustrate what we're doing, just because that's an easy year to look at. So to estimate the population size, we are going to calculate the population index, which is just the area under that curve, which in this case is about 27,000. This is measured in butterfly days, not in butterflies, because what's observed are active butterflies. So it is proportional to the population size, but it's not an exact count of the population. So that's pretty straightforward. Phenology is a little bit more complicated to talk about because there are a lot of aspects of phenology we might want to measure. So I'm going to talk about the 3 that have been used in butterfly studies in general. And I feel like they kind of cover the gamut of things we might be interested in. So we have the onset. So when does the activity start? And historically, this tended to be measured by what was the day that we first saw butterflies. That actually has been shown to be kind of problematic in that it changes based on population size, even if the underlying behavior isn't changing. And so we're using the quantile approach. So we're defining the onset as the day that separates the 10% of the earliest activity from the 90% that's later. And in this case, that happens to be day 235. Instead of just being interested in that sort of beginning of activity, we might also want some measure of the average activity of the population. We're going to use the mean day of activity here. This is a Gaussian curve. That's very easy to define. It's the peak, it's the median. Gaussian curves are fantastic. In this case, it happens to be day 243. And then the last piece that has been important, was particularly important in the McKlin study, was the flight period. So this is a measure of how long are the individuals active. And we've actually seen some other talks in this session that have been talking about how that might matter. So we already have our measure of onset, which is based on quantiles. We can take the same approach to define an end of the activity. And that defines a region in which 80% of the activity is happening. And we can just count, well, how many days or parts of days separate those 2 points. So how long are they active in general? If that number gets smaller, we're talking about a tighter window of activity, and that'll have consequences for the way they use resources and the way they interact with predators. All right, so we've got our 4 metrics, and we can actually take our data set. I've been showing the ridges plot just for the most recent 10 years, but we have a longer data set for the Lang's metal mark. So in terms of population index, we get something that looks like this. I'm putting it on a log scale so that the slope of each of those lines corresponds to growth rate. And we can see, like we saw with the actual curves, that the population was increasing early on, but seems to have decreased in recent years. We can also calculate all of our phenology metrics. So in this case, it looks like they're shifting to be active on average, maybe a little bit later during the year, based on the day of onset and on the mean day of activity. And the flight period looks like it might be increasing a little bit. I should point out that because the Gaussian curve is symmetrical, for any 2 of our 3 phenology metrics, if we have them, we can define the 3rd one. I'm going to keep showing you the results from all of them, though, because you shouldn't have to calculate that in your head. So, all right, so we have a bunch of species and a bunch of sites, and I showed you the plots for one site. The overall result across all of these species is that they are, on average, declining a little bit. This is disappointing, maybe, but not at all surprising, because remember, these species were selected specifically because we're concerned about them. These are species that we think might be declining. We're also seeing changes in phenology. It looks like, on average, they are becoming active sooner in the year, and our flight period looks like it's shrinking. So we have this tightening window that is shifting to be a little bit earlier on average. But I will tell you there's a lot of variation. So neither of these seems like necessarily very good things, but do we see that same pattern of interaction that McKlin and McGregor saw, where the shifts in phenology are mitigating population level problems? And I even wrote that down. And the answer is yes. So even though these are univoltine species, we're seeing that the advancing onset, so negative values on the x-axis, correspond to populations that seem to be growing on average, and that the populations that are shifting to be active later in the year instead, they seem to be doing poorly. We see that with our measure of onset, and we also see that with our measure of peak or average activity. Now, the McKlin paper found that the flight period also really mattered, but that largely corresponded to multivoltine species. And so it was really interesting to us that we found that same positive relationship. So in general, mostly we're seeing it sort of to the left of 0. So mostly the populations that have shrinking flight periods, so shorter periods of activity, also seemed to be the populations that were declining. So this is really exciting. Even in these at-risk univoltine butterflies, where we didn't have a lot of information of what was going to go on, it seems like tracking the climate might be good for the populations.
4 minutes.
Perfect, thank you. I should point out, this is correlation. So like all the other studies on this, all that we can say is that those populations that are active earlier seem to be growing on average. There's a lot more that we can do with all of this fitted data. We have abundance trends and we have phenology trends for each of our sites. And so I'm really excited to do some other work with them, to link those trends to butterfly traits. So the McGregor paper found that host plant specialization really influenced the relationship between abundance trends and phenology. And there are also a lot of butterfly attributes that seem like they should be important for phenology and maybe also for population growth. Similarly, we know the information about those sites. So in the McKlin paper, the relationship between abundance and phenology depended in part on whether those butterflies were at the northern or southernmost edge of their range. The other piece to this is that these species were selected specifically because they're of conservation concern. And we're really excited to take information on how the sites have been managed and look at the population level consequences for those butterflies. This should be useful for basic science, but also hopefully will provide some useful tools for land managers of what management is working and what is not. With that, our work was funded through the CERTIP grant. June Arians and Christopher Jason were incredibly helpful in this work. I've got some sources for the photos. I should point out, I did not gather 800 years worth of site data. And instead, we have a lot of sources of this data. And I would be happy to take any questions.
Great, thanks, Collin. So again, you can use multiple ways to get questions to Collin. You can use the Zoom window or Slack, whatever works. So we have a question in the chat. Great talk. So your statistical approach assumes that phenological distributions are unimodal and follow one single Gaussian distribution. I think that's great, but phenologies, oh man. There are other taxes and phenologies which don't show this distribution, so maybe they're multimodal or asymmetric. What methods would you suggest for those phenological distributions?
Yeah, so I am pointing to, so we were specifically interested in univoltine species, but also, like you say, this method does not work for bivoltine or multivoltine species. There are some really good papers recently, so like Tyson Weprick and Stemkowski both have really good papers using generalized additive models. Those are flexible and do a good job of fitting unimodal data. In general, the more flexible you need your method to be, the more data you need. We had quite a few years of data where we had 4 or 5 non-zero observations. And those are just really hard to fit with anything flexible. For the Carner's blue that I'm showing here, there's a lot of data, and this could be fit with a more flexible approach. For the multimodal, I really think that generalized additive models are probably the best approach. Yeah.
Great. There's another question in the Slack. I don't know that we have time for it. Sasha, what do you think?
Maybe not this time.
Okay. All right, so just check the Slack channel, Collin.
Sounds good. Thank you.
Okay, so our next speaker this afternoon is Stephanie Varghese, and she'll be presenting a talk entitled Silica Gel Packets as a Novel Desiccation Strategy for Enhancing Plant Drought Experiments.
Hi, my name is Stephanie Varghese, and I'm an undergraduate researcher of plant ecology at the California State University of Los Angeles. I am extremely grateful for the opportunity to share with you today a novel dehumidification strategy for enhancing plant drought experiments. As global temperatures rise, the frequency and severity of extreme climate events such as drought are escalating. Contemporary drought experiments simulate natural drought by reducing soil moisture via controlled watering regimes and rainout shelters, 2 of which are shown here on the right. While such practices can effectively simulate drought conditions at the soil level, they can be ineffective or sometimes counterproductive when it comes to simulating drought at the atmospheric level. For example, it has been shown that rainout shelters can actually increase ambient air humidity by impeding wind flow and thereby restricting ventilation. By manipulating atmospheric drought in addition to soil drought, we can better predict plant community responses to drought. Plants need water to perform basic metabolism, and the relative water balance within a plant is the result of not just water availability at the soil level, but also the rate of water loss at the leaf surface. For example, plants reduce stomata conductance to regulate transpiration. In the context of drought, plants reduce stomatal pore aperture in response to both humidity deficits and soil moisture shortages. In doing so, plants are able to maintain hydration even when conditions are very dry. But at the same time, this can result in decreased carbon fixation and hence community productivity. With this in mind, evaluating the interplay between water scarcity at both levels is central to developing our understandings of how plant communities respond to drought overall. This figure shown here comes from a recently accepted article by Aguirre and Wright et al., also from our lab. It demonstrates that changes in atmospheric moisture can significantly affect biomass, even if soil moisture is kept consistent at a drought level. Our lab's biodiversity drought experiment simulates drought at both the soil and the atmospheric levels. In the top two photos, we see what our experiment looks like on a typical sunny day in California. And in the bottom photo, we see what our experiment looks like on a rainy day when we have our rainout shelter in place. This particular study focuses on data from the winter and spring 2020 growing season, which took place between January 2nd and May 12th of the year 2020. Of our 66 mesocosms assigned 3 grass biodiversity levels, 2 soil moisture levels, and 3 microclimate air humidity levels, we were particularly interested in our 6 bare ground pots that were assigned our dehumidification treatment. This first image on the left shows what one of those 6 pots looks like in person. And here is a breakdown of that pot setup. We monitor relative humidity at the microclimate and macroclimate levels using iButton data loggers, which are programmed to take continuous readings every 5 to 10 minutes. And we use silica gel packets as a practical, low-cost, and effective means of regulating microclimate humidity. Silica gel itself is a solid, granular form of silicon dioxide with a massive internal lattice of interconnecting microscopic pores. This material is capable of absorbing water vapor from the environment and retaining that moisture via a process known as capillary condensation. Packets continue to desiccate in this way until an equilibrium with the relative humidity of the surrounding air is achieved. Once saturated, silica gel packets can be reheated in a microwave oven to drive off excess moisture and then be reused in the experiment. Silica gel desiccation is a passive process driven by concentration gradients. When a packet has a lower internal water concentration than the surrounding air, it can effectively dehumidify the air. But this process might also work the other way around. When a packet has a higher internal water concentration than the surrounding air, it may actually emit humidity. Since that second case is the opposite of what we would want to simulate in our drought treatments, we wanted to make sure that we understood the different factors that may activate it. Likewise, our first objective was to quantify the efficiency of our desiccation treatment under various environmental and experimental conditions, such as macroclimate humidity, soil moisture, time of day, and the frequency of packet replacement. Our second objective was to investigate how silica packets can augment the moisture manipulation methods that are common in current drought experiments by desiccating effectively alongside rainout shelters and drought watering regimes. And our third objective was to develop practical advice for researchers who might be interested in adopting our methods. From our iButton data, we calculated humidity averages over 12-hour periods, 24-hour periods, and single-hour periods, each comprised its own data set. We decided to build our main model around the daytime-only data set in order to highlight how effective this desiccation treatment is during the time of day when key physiological measurements such as stomatal conductance are being measured. The other data sets were used to answer secondary questions. So, for example, how the degree of dehumidification depends upon fine scale changes over the course of a day, in which case we consulted our hourly dataset. In each model, our response variable is the microclimate relative humidity effect, which tells us whether the humidity in a desiccated pot is in fact lower than that of a control pot, and if so, to what degree. We calculated this as the difference between the average macroclimate and microclimate humidity levels, or in other words, the humidity in a pot with packets minus the humidity in a pot without packets. Our first hypothesis is that the degree of daytime drying by silica gel packets is affected by air humidity, soil moisture, and frequency of packet replacement. To explore that first hypothesis, we first examined how ambient air humidity affects the strength of the desiccation effect. On the y-axis, we have that microclimate RH effect. When y is less than 0, the packets are effectively desiccating the microclimate. When y is greater than 0, the packets are emitting humidity into the microclimate. And when y equals 0 is when packets are at that threshold of neither emitting humidity nor dehumidifying. Many of these points fall below the y=0 mark, which tells us that packets were working effectively for the most part. This was true even on rainy days when our rainout shelter was in place, as indicated by these X-shaped data points. We also found that the desiccation effect was significantly stronger on humid days. This tells us that packets are more likely to exhibit that adverse humidification effect when it's dry out. But more importantly, they dehumidify the air efficiently when it's humid out, as is the case on rainy days when a rainout shelter is in place. Thus, putting out packets on a rainy day might be an effective way of reducing relative humidity to more closely simulate drought in those types of experiments. So now that we have a better understanding of how moisture at the air level affects packet performance, what happens when we add the component of soil moisture? This next figure illustrates just that, the interaction between macroclimate humidity and watering regime. In our pots that received ambient watering, as shown by the blue line, we see the same trend in the desiccation effect. Packets dehumidified the microclimate more effectively when there was a higher amount of moisture in the air. We see a slightly different trend in our drought water pots, as indicated by the red line, where packets dehumidified the microclimate effectively, regardless of macroclimate humidity. This tells us that packets reduced air moisture well when combined with this watering treatment designed to reduce soil moisture, which is another key manipulation in plant drought experiments. And here is that same figure side by side with the main effect for soil moisture to help illustrate that last point. Note that packets might be working less effectively in pots of ambient watering because they increase the rate of evaporation off the soil surface, saturate with that soil moisture, and then might have less space to accommodate for air moisture as a result. And in this next figure, we see how leaving packets out for a long time can actually weaken their dehumidification ability. When we leave packets out for a long time, they might saturate, reach their adsorption threshold, and then stop desiccating. And in some cases, saturated packets might be found to re-emit humidity into the microclimate. We don't want that to happen in our drought treatment pots, which is why it's essential that we replace packets every so often. When COVID restrictions limited our ability to access our experiment, we were still able to keep our drought pots dehumidified by changing packets every 2 days, which is why we have a good amount of replicates up to there. But based on this trend line, we may have even been able to get away with changing them every 3 to 4 days as well. Our second hypothesis is that the nighttime dehumidification effects in California are stronger than the daytime effects. To explore that hypothesis, we examine the main effect of temporal period on the desiccation effect. It's important to note that California's climate consists of hot, dry days and cool, humid nights. So it makes sense why the desiccation effect is stronger at nighttime. Packets work better when there's a higher content of moisture in the air, and in our climate, that is the case at nighttime. Our third hypothesis is that packet efficacy is dependent on lagged effects from the previous day's conditions. To test that hypothesis, we explored our 24-hour model's ANOVA table results for the interaction between the macroclimate humidity levels of a current day and of a previous day. But we found this interaction to be non-significant, indicating that the relative humidity of a previous day does not significantly impact the desiccation effect on the present day. This tells us that even if a packet was absorbing a huge amount of moisture on a humid day, it can still have a chance at working as effectively as usual on the following day. Our fourth hypothesis is that the degree of dehumidification depends upon fine-scale changes over the course of a day. This figure illustrates the hourly desiccation effect on days of different levels of ambient air humidity. The light blue lines represent the degrees of packet desiccation, and the dark blue lines represent macroclimate humidity. We separated this figure into 3 distinct panels so that it's easy to see how the hourly desiccation effect might vary in climates characterized by low, medium, and high relative humidity. In each panel, the light blue microclimate RH effect line almost exactly mirrors the dark blue macroclimate RH line, demonstrating that packets respond fairly quickly and throughout the 24-hour day to changes in air humidity. In other words, we don't see much evidence that packets require a whole lot of time to adapt to changes in air humidity. Around 8 to 10 a.m. in each panel, we see a significant dip in the desiccation effect. This might have to do with packets' desiccation ability being impaired when they've been absorbing air moisture effectively for quite some time. Overnight and then early the following day, there's a huge amount of moisture in the air for packets to absorb. When the air moisture level starts to drop at 8 a.m., the saturated packets might emit some of the previous night's humidity and then go back to desiccating effectively at 9 a.m. until the air humidity decreases at 10 a.m. and then continues to drop through the remainder of the day. In light of these findings, we found that silica gel packets work as effective atmospheric desiccants under a variety of conditions, especially those employed in contemporary plant drought experiments. More specifically, we found evidence that packets work least effectively when the air is dry and the soil is wet, which may be because the moisture gradient causes packets to re-emit humidity rather than dehumidify. But we also found evidence that packets work most effectively when the air is wet and the soil is dry, which, importantly, is the exact combination that we see in experiments that simulate drought through rainout shelters and through drought watering regimes. If we take these principles and walk through the course of a 24-hour day, it becomes clear how packet saturation might impact packet efficacy. Note that the trend we're about to see in the next figure is largely mediated by region-specific effects. More specifically, California's climate consisting of hot, dry days and cool, humid nights. Overnight, packets reach the highest point of saturation, but when air moisture begins to drop at 8 a.m., packets may begin re-emitting the previous night's humidity. By 9 a.m., the packets may have emitted enough moisture to be dry again, and therefore work as effective dehumidifiers again. But as humidity continues to drop at 10 a.m., and then throughout the remainder of the day, packets may either absorb or emit humidity, depending on the moisture content on either side of the air packet equilibrium. So to summarize these findings, we found that packets desiccate most effectively when the atmosphere contains more moisture than the packets. This is true when the macroclimate is humid, when it's nighttime in California, and when a rainout shelter is in place. Similarly, we found that packets are more likely to counterproductively humidify the air when they contain a higher moisture content than the surrounding air. This can be the case when they saturate with soil moisture or when they haven't been replaced in several days. And finally, we found that packets can enhance drought studies by absorbing excess humidity produced by rainout shelters and by reducing air humidity to drought levels on particularly wet days, which is when the ambient weather conditions are least similar to what we would want to simulate in a drought experiment, and an effective desiccation treatment is therefore most needed. Here are my references. And with that, I would like to thank my advisor, Dr. Wright, my thesis committee, the MORE programs of Cal State LA, and everyone in the Wright Lab who helped to make this experiment possible. And I would also like to thank ASN for the opportunity to share this with you today. And lastly, if you're interested in discussing any of our methods or findings in greater detail, please feel free to reach out to me or my PI. We would be more than happy to connect with you and answer any questions you may have.
Great, thanks, Stephanie. For anyone who has questions for Stephanie, please submit them to me via the Zoom chat or through Slack. We have about 5 minutes for questions. Great, so we have a question. It seemed as though the reduction in humidity was a few percent. Is that right? Is this an amount that mimics the biological conditions you're interested in?
I did want to mention that I'm actually having issues with my audio today. Would you be able to paste that question into the chat?
Yeah, I can. You should have it now.
Yes, just got it. Okay, that's a really great question. The minor changes that we found in humidity are largely due to California's climate. We are primarily hot and dry, so we didn't have a lot of replicates of very drastic changes in, like, these are dry days and these are humid days, which is why we see those such slight fluctuations in our model. But ideally, we would like to have other drought experiments in different climates replicate our experiment so that we would have a better understanding of how well this desiccation treatment might work in climates that have more drastic fluctuations in humidity.
Okay, I'm going to send you another question in the chat, and then I'll just say it so everyone can hear it. So the question is, how do you think this would work on a larger scale?
Another great question. As a reminder, in our experiment, our study system focused on drought simulation at a very small level. We have mesocosms, which are essentially pots with like a covered outside. But if we wanted to repeat our experiment in a larger setting, like a field setting, we would probably want to set up some type of free air dehumidification system. So in that case, silica packets might not be the best choice. We might want to actually rely on industrial dehumidifiers because those would probably work a lot more effectively on a larger scale. But I think the most important component is that an effective atmospheric desiccant is applied in these types of drought experiments.
Okay, Scott, are there any other final questions or should we move on?
No, we can move on. If anyone has other questions for Stephanie, feel free to send them to me and I can send them to her through Zoom.
Great, thank you so much.
Thanks for that great talk, Stephanie. So our next talk today is from Alec Chiono, and they will be presenting a talk entitled Testing the Climate Variability Hypothesis with Coastal and Inland Populations of Erythrantha guttata.
Yeah, or Mimulus guttatus, which is what I have up there now.
Oh, great.
Thank you for the introduction. Yes, my name is Alec Chiono. I'm a master's student from the University of San Francisco. Excited to talk to you today about my research. I actually finished collecting these data a couple weeks ago, and I have some surprising results, but that's just to say these analyses are preliminary, so I really welcome any feedback you might have on what I'm sharing with you today. So my project about the coast starts with this paper about the tropics. So some of you might be familiar with this work by Dan Jansen. And in it, he talks about how tropical organisms experience a smaller range of temperatures than temperate organisms usually do. So you can see that on these plots of temperature across the year. There's obviously very little fluctuation in temperature at that tropical site. And so we might expect that tropical organisms are able to handle a smaller range of conditions than temperate organisms. And Jansen was interested really in taking this idea and extending it, thinking about mountain passes as ecological barriers. But this underlying idea about temperature variation and thermal niches kind of took hold from his paper as well. It's become known as the climatic variability hypothesis. Simply put, it's that organisms in more variable climates should be able to handle a wider range of climatic conditions. And there's been a decent amount of follow-up work testing the climatic variability hypothesis. And most of this has been in the context Jansen was thinking about tropical and temperate organisms. And generally there's been support for this. So you can see on the graph there that these tropical stream insects both experience a smaller range of temperatures and also can handle a smaller range of temperatures than temperate stream insects. But we can apply this hypothesis more broadly, test it more broadly. And coincidentally, Jansen noted in his original paper that the coast around the San Francisco Bay Area is an area of great temperature stability. And this is true. So you can see temperature data for 3 of my study sites. And that coastal site, you can see there's very little fluctuation, both annually and diurnally. And you only have to go a few kilometers inland before those fluctuations really start to change. And as you go more inland, those fluctuations generally get larger. And so according to the climatic variability hypothesis, we would expect that coastal organisms should be able to handle a smaller range of conditions than inland organisms. And so that's my big question. What I'll be talking to you about today is if coastal plants have smaller thermal niches than inland plants. And so what is a thermal niche? It's the set of temperatures under which an organism can persist. And there's a lot of ways we can try to measure this for different kinds of organisms. But for plants, it's pretty common to measure growth across a range of temperatures. And then you use that data to build a performance curve like you can see in the schematic there. And then you can measure the niche as the width of that curve. And that's what I did for my project. So my study species is Mimulus guttatus or Erythranthe guttata and grandis. So it has coastal and inland populations. And when I say coastal, I mean really super coastal, growing on bluffs and seeps right next to the beach, meters away from the ocean. And an important note here, the species is known for having annual and perennial ecotypes, but I used only perennial populations for my study because I'm thinking about temperature variation across the year. And so I want organisms that are around to experience that full variation. So in the summer of 2019, I surveyed a number of sites looking for populations that experienced a range of temperatures, but also had plenty of plants to collect from. And so I focused on 6 populations. You can see that there are 3 coastal ones in the San Francisco Bay Area and 3 inland ones with varying distances from the coast. But of course, a population being coastal or inland doesn't tell us exactly how much temperature variation it experiences. So I retrieved downscale climate data for the last 40 years using the niche map R microclimate packages. But I also wanted to verify this data since microclimates can vary quite a bit. And so I set up temperature loggers in the field at my study sites, 2 at each site, to actually see what temperatures are going on out there. And the nice thing is that these data sets are both reported hourly, so I can compare them pretty easily. And so here's a graph of the temperature variation at each site for the downscale data and the HOBO data across the same time period. You can see it's about a year up there. And it's actually pretty great that downscale climate data captures the temperature variation pretty well, except for one site, Point Reyes, seems to be underestimating that temperature variation a little bit. But now that we know the downscale climate data does a pretty good job, we can look at that data set for the last 40 years. So we know what these plants have been experiencing. In the longer term, and I'll put a little arrow there to show that we think Point Reyes is probably a little higher. But the more important thing here is that these populations span a range of temperature variation. And also interestingly, there's some differences within both of our coastal and inland groups here. So once I had collected my field seed, I was ready to perform controlled crosses. So you can see the number of seed families I was able to generate there. And so I randomly crossed within each population to produce full sibling families with unique parents. Once I had that cross seed, I was ready for the actual thermal performance experiment. So the important things to note here are that the plants were only a few weeks old when I moved them into the growth chamber for their 1-week temperature treatment at their designated temperature. So you can see the range of temperature treatment there. And I took photos before and after to measure leaf area and use that to measure growth. And so you can see on these 2 trays on the right here, these are the same tray before and after treatment. They can actually put on quite a bit of leaf area during that time. And so I can calculate the relative growth rate of each individual as the difference in their leaf area divided by the number of days, and all of that weighted by the size of the plant at the beginning of the treatment. And a really important thing to say here is that this is not a measure of fitness, but it is performance at a critical life history phase. So this allows us to still build some relevant thermal performance curves. So data processing, the fun part. So once I had all those pictures of the trays, I used the OpenImageR package in R to programmatically crop each individual with its respective scale for size. And then I used the program Easy Leaf Area to actually measure the leaf area of all those individuals. And then with that leaf area data, I was able to calculate the relative growth rate for everyone. And then I used the PerformR package in R to build my performance curve. So this is a hierarchical Bayesian model specifically meant for building performance curves. And I just really have to thank all of the authors of these programs, because given the craziness this last year, restricted access and everything, all of these methods really made my master's thesis a lot more feasible. So here's the sample size. So you can see the numbers there, a little over 2,000 individuals total. But before that data go into the PerformR model, I average by family within each treatment. So that gave me a sample size of 613. And now we can talk about the results. So here are the performance curves. So I'll orient you to this graph. So you can see on the x-axis is the range of temperatures and temperature treatments marked there. And the y-axis is the relative growth rate. And then each curve is colored by its respective population, with coastal populations being variations of the color blue and inland populations being variations of the color orange. And so each of these curves tells us how well that population grew at that given temperature. But I want to compare the niche breadth of all these curves, right? So you can see these dashed lines representing niche breadth at different locations on those curves. So I measured the niche breadth of each curve at 50% of the maximum relative growth rate and at 80% of the maximum relative growth rate. I wanted to make sure that both of these measurements told the same story. They do, they're qualitatively similar. So I'll just show you data for breadth at 80%. Now this is what we might expect this niche breadth data to look like. So as we move more inland to more thermally variable sites, we would expect the niche breadth to generally increase. And so I'll fade that into the background for your reference. And here are the actual data. So you can see this is a little bit surprising. Mono Lake, our most inland population, actually had the lowest mean niche breadth. Chimney Rock, our most coastal population, had the second highest mean niche breadth. But I need to put the 95% credible intervals on there. And we can see they're all really overlapping. And so I can't differentiate in the niche breadth of any of these populations. But of course, as I mentioned, whether something is coastal or not doesn't tell us what kind of temperature variation it's dealing with. So I can plot the niche breadth against the actual temperature variation of each population. Again, you can see those grayed out dots in the background showing we would expect a positive trend here. And unsurprisingly, given the last plot, there's not really any differentiation here either. And so it seems like these plants, despite experiencing a lot of this temperature variation, they really just kind of like to grow at about the same temperatures. And so I found no support for the climatic variability hypothesis, but there was an interesting difference in another growth parameter. That was the maximum relative growth rate. So you can see here another schematic. So that dashed line is representing the maximum relative growth rate. And so that is basically the how fast the plant can grow at its ideal temperature. And again, here's a plot with expected data. So those inland sites are more seasonal in temperature, as I've shown you, but they generally tend to be more seasonal in their water availability. And so they tend to have shorter growing seasons. And if they have shorter growing seasons, we might expect them to, try to grow faster when conditions are right. So again, I'll fade that into the background. And here are the actual data. So interestingly, we don't see differences generally between coastal and inland sites, but we actually see differences within the inland population. So you can see Mount Diablo there has a lower maximum relative growth rate than the other two inland sites. And this was kind of surprising to me at first. I still don't really know why, but I have a clue maybe from how the plants survived at the highest temperature treatment, the 40 degrees. So there was a decent amount of mortality. Here you can see some of those coastal populations did not do so well, but Mount Diablo did the best of any populations. And so potentially there might be a trade-off between its ability to handle these temperature extremes and how fast it can actually grow. But at this point, it's really just a hunch. So in summary, I found no support for the climatic variability hypothesis. And then there was unexpected differentiation in the maximum relative growth rate. And lastly, I just want to talk about how these performance curves may not tell us everything about thermal niches. So of course, I've been talking about thermal variation across the year, but generally for plants, there's a time of year they like to grow. And so the temperatures during that time might tell us a little bit better what the plants, what temperatures they like to grow at. And those might not be so different between all of these sites. And of course, another important thing here, as I mentioned, this is growth at an early life stage. It isn't fitness. And so there might be a disconnect between what I have here and what their actual fitness is. So I'm following up this experiment. I'm going to measure fecundity on a subset of individuals from the experiment and see how predictive relative growth rate is of fecundity. And kind of on another side, I'm going to use this thermal performance data to try to forecast how suitable thermal habitat might change for all of these populations. So initially, I was thinking coastal plants might have smaller thermal niches, and they might be more sensitive to temperature increases from climate change. Of course, not really expecting to see that now, but I have all of this great thermal performance data, and so it will be interesting to see how suitable habitat changes for all of these populations as climate change proceeds. And so with that, I'd like to acknowledge everyone that made this research possible, all the people that gave me help and advice, of course, collection permits to actually get the seeds, and my funding sources. And with that, I'll take questions.
Great, thanks, Alec. So we have a question for you from Megan. Thanks for the great talk. I'm wondering if the niche breadth of your Mimulus species is relatively large or small compared to other species across the study areas. Also, what relationship do you expect between the species' niche breadth and competitive ability or overall survival across these coastal to inland populations? And I'll also send this to you in the chat.
Yeah. I already forgot what the first question was.
Okay, the first part is whether if the niche breadth of your Mimulus species is relatively large or small compared to other species across the study areas.
Yeah, okay, so that's a good question. Yeah, I can't say that for sure because there hasn't been a lot of like great plant coastal thermal niche studies, but my expectation would be is that this Mimulus, it is a widespread species that maybe hasn't been isolated from these inland populations all that long. And of course, there are some coastal and lineages that have been isolated for longer periods of time. So they might have evolved smaller niche breadths, but yeah, I can't say for sure about that. But yeah, it would be interesting to think about inland too, how that compares as well. I imagine they're probably more similar, but I'll have to look at that. That's a really good question. And then for the next question, yeah, I think that's a pretty interesting question too. And thinking about, yeah, kind of Yeah, I guess this is another one where it's kind of hard for me to say because there are a lot of these different factors. Of course, you might think about the maximum relative growth rate being important for competitive ability, and whereas niche breath, if it's larger, then you might not have to deal with competitors as much. Yeah, I'm not sure if I guess maybe what I should say is I'm not sure what kind of relationship I would expect between that, especially not being able to comment on what the niche breath is of all the other plants that they're competing against in these locations. But both really good questions and something for me to really think about.
Great, we have a few more questions for you. I think there's time for some of them. Awesome talk. I lost a minute there, but do you think Mount Diablo populations have historically experienced higher temperatures than the other sites?
Yes, so that downscale climate data actually tells us no, but growing up here in the Bay Area, Mount Diablo is called Mount Diablo for a reason. So I was a little bit surprised by that climate data, basically, in short. And so I kind of have this that climate data seems different, but my personal hunch is that I actually think they are dealing with hotter temperatures than some of these other sites. And the other thing that's I guess might play a role into that's harder to measure is how much water availability all of these plants really have, and that of course I think might be lower at Mount Diablo for these other ones, and so they're really kind of being stressed out by these warmer temperatures, or would be, but have perhaps adapted to handle them better.
Great. One more quick question, and I'll post some other ones to you because there's a bunch we can't get to. Great talk. Are you going to use any other climate variables from these different sites other than temperature, maybe rainfall or water availability?
Yeah, I'll definitely look into that, but we originally just kind of didn't worry too much about water availability because Mamilos grows in these seeps where water just collects. And so obviously water is super important for this plant. It likes a lot of it, but it's kind of hard to say with accuracy what, because they happen to be growing in these seeps, and what area happens to be a seep and has all that water available to it.
Great, thanks, Alex. So I'll send you a couple more chats in Zoom, and there's one more to check out on the Zoom or on the Slack channel.
Thank you.
Okay, great. Awesome talk, Alec. So our next speaker is Emelin Resiteritz, and she'll be presenting a talk entitled The Influence of Temperature and Nutrients on Disease. Go for it.
Need to unmute myself before I share my screen and appears. Okay. All right, can everybody hear me?
Yes.
Okay, perfect. All right, so the title's changed a little bit, but I'm going to be talking about the influence of temperature on host parasite dynamics. So thanks for coming to my talk. I'm currently a postdoctoral scholar at the Center for the Ecology of Infectious Diseases at the Odum School of Ecology. It's a long name, it's a mouthful. But today I'll be talking about some of my recent postdoctoral work studying the influence of temperature on host parasite dynamics. So I won't belabor the point, especially since we're in a climate change section, but global climate change, especially rising temperatures, will likely have impacts on host parasite dynamics. And in order to understand how parasites will be impacted by climate change, we really need to understand how temperature influences the host itself, how temperature influences the parasite, especially the free living stages. And then we need to understand how temperature influences the interaction between host and parasite. So for instance, we need to understand how infected hosts might respond differently to elevated temperatures than uninfected hosts. Now, very little work has been done on understanding how complex life cycle parasites will be affected by climate change. And so that just adds a whole other level of complexity. So in order to understand the effect of temperature on complex life cycle parasites that have these multiple hosts, we must understand how temperature affects all of the hosts, both definitive and intermediate, as well as how temperature influences the interactions between this parasite and each of these hosts. So yeah, the green represents all the hosts, the red represents the free living parasite stages, and then those black arrows are the interactions between those hosts and parasites. So for the work I'll be talking about today, I'll be focusing on the first intermediate host, the ram's horn snail, of a variety of different trematode parasites. So important to note is that when ram's horn snails are infected by a trematode parasite, the parasite forms a colony in their gonads, thus castrating the snail host. Because ectotherms, such as snails, are highly influenced by temperature, temperature is likely to have large impacts on these host parasite dynamics at this life stage. So the work I'm going to be talking about today was done in collaboration with Rebecca Atkins, a PhD candidate in my department, as well as Jason Ogbomo and Will Ellis, who are both undergraduate researchers. So for this project, I worked in the Huey constructed treatment wetlands, which are south of Atlanta. And each of these wetlands, shown in these different colors, are made up of multiple different cells and are all fed the same treated wastewater. So these different cells within a wetland are connected such that warm treated water flows sequentially through each cell from 1 to 2 to 3, which creates this potential gradient of both nutrients and temperature, which is replicated across these different wetlands. So it's a great experimental system that I could never afford to recreate. So the first thing we wanted to understand was how temperature influenced the snail host in and of itself. So in order to do this, we collected snails from 3 different wetland cells at Huey, the wetlands, and allowed these snails to lay eggs in the lab over a 3-day period. Very quickly, they laid a bunch of eggs. They were very excited. These eggs were then reared in the lab, creating a cohort of genetically similar uninfected snails. Now, once these snails were large enough, they were painted based on wetland cell origin for the experiment, with the idea that snails might respond differently to temperature based on the wetland origin if there are these temperature differences that we expect. So we placed individually marked snails across 5 temperature treatments. Each replicate had 3 snails, 1 from each wetland, and we had 7 replicates per temperature. Now, individually marked snails, we did this because it allowed us to measure growth and mortality on an individual basis. And while ram's horn snails are hermaphroditic, they don't readily self-fertilize, and so it was important to have multiple snails per replicate. So we also were able to measure fecundity, or the number of egg clutches produced for each replicate, though we couldn't identify which of the 3 snails in that jar were actually producing those eggs. So we ran this experiment for like 6 months, but I'm only going to show data for the first about 10 weeks. So after 10 weeks, we measured the performance of snails actually every 2 weeks. But after 10 weeks, we found that the survival was highest at about 12 degrees Celsius, though there's not really much of a difference between our 12 and 20 degrees Celsius treatments. And we also found that snails grew fastest at 20 degrees Celsius. And anything above that, dramatically, you got a decrease in growth. Additionally, we found that our snails reached sexual maturity earlier and at a smaller size under these warmer conditions. And we determined sexual maturity as the first date that there was an egg mass in a replicate. And this relationship that we see, known as the temperature size rule, has been used to explain why ectotherms at northern latitudes are often larger than in southern latitudes. But despite having the warmer snails in the warmer treatments maturing faster, T4 in yellow, this warmer treatment, had significantly lower fecundity over the duration of the experiment than T3, which was our more moderate temperature. All right. Next, we wanted to determine if temperature influences parasite transmission. So for this, we focused on the free living stage of the parasite between the final host, which can be birds, mammals, or fish. We're not sure in this case. And the first intermediate host, which is the ram's horn snail. So at this point, we can't replicate the full life cycle of these trematode parasites in the lab. So in order to look at transmission from final hosts to our snail first intermediate host, we have to do these field manipulations or surveys. So we used our genetically similar uninfected lab-reared snail cohort, the same cohort of snails from experiment 1, but not the same snails that we used. So half of the snails went to experiment 1, and then half of them went for this field experiment. And we individually marked 15 snails per cage, 5 from each wetland cell, and cages were these modified minnow traps. We then placed these cages across 3 wetlands, and the first 3 cells per wetland for a total of 9 wetland cells marked here. Because water comes in hot from the plant at the first cell, number 1, we believed that there would be a temperature and also a potential nutrient gradient across these wetland cells. So this design allowed us to have essentially 3 replicate gradients. And we also took nutrient samples monthly and deployed a hobo data logger within one of the cages for each of our wetland cells. We also had camera trap data. So we had a camera on each of the, at each of the sites. And because these snails are genetically similar and are the same age and reared in the same environment, this allows us to have a really highly controlled field experiment where we can assess differential rates of parasitization in the field across repeated temperature gradients. These snails were in the field for 100 days, which nicely coincided during the part of the pandemic when I was not allowed in the lab at all. So we put them out and they did their thing while we stayed at home. And these snails were then brought back to the lab after 100 days and placed in individual containers. Now, over 4 weeks, snails were shed, which means that they were exposed to light and heat to induce parasites to release their free swimming stages. So if Cercaria were present, which you can see here with the snails emerging, these little dots are all Cercaria that are emerging from an infected snail. So if we saw that, we could mark these snails as infected. Remember that these parasites castrate their snails. So on the other hand, if snails can store sperm past the time when they're actually interacting with another snail. So uninfected snails that had their gonads intact could produce egg masses even in isolation. And so snails with egg masses were then marked as uninfected. So we could easily identify snails that were definitely infected and snails that were definitely uninfected. So we put 250 snails out in the field. We got about 130 snails that survived being out in the field for those 100 days. And of those, 33 of those snails were infected. So we found 33 snails infected from 2 different species of trematode. We don't know the actual species identity at this moment, so we're unsure of what the secondary and final host species are. These are really early stages of trying to set up this system. So when we look at infection prevalence, or the proportion of snails that became infected during these 100 days out in the wetlands, we found a really strong relationship between cell number and infection prevalence. So while cell number isn't a continuous variable, I'm applying it as one for ease of seeing this relationship. So at cell 1, there were no infections. These are all 0 points. They're just jittered. At cell 2, there was a mix of infection levels from 0 to 30%. And then cell 3, infection prevalence was consistently above 40, mostly 50% infection prevalence. So half of the snails at wetland cell 3 were infected with this parasite, which means half of the snails were castrated. Now, originally we had set up this experiment, we had hypothesized that there would be a temperature gradient across these wetland cells, and that's what we were going to be looking at. However, the HOBO data suggests that temperature does not explain the relationship between wetland cell number and prevalence. However, the HOBO was data logging temperatures around the edge of these wetlands where the snails live and where our exclusion or where our cages were. And these temperatures, while they give us the microhabitat that the snails live in, may not represent the full range of temperatures that the trematode eggs / Miracidia actually experience on their search for their snail hosts. So we may not be getting the full picture of how temperature might be affecting transmission. However, the relationship between infection prevalence and wetland cell was very strong. And so we looked at a variety of nutrients as well as camera trap data to determine bird activity and diversity to see if any of these variables might explain the strong relationship we found across wetland cells. We found a significant positive relationship between bird activity and infection prevalence, suggesting that final host distributions are dictating infection prevalence rather than environmental variables that influence the parasite itself. And this also gives us some hint that the final hosts of these trematode species may be birds, but it's not conclusive. We also found a nearly significant negative relationship between ammonia concentration and infection prevalence. In general, these wetland cell 1 had high levels of ammonia compared with 2 and 3, but remember the water has already been treated and is drinkable at this point. So the levels of ammonia are pretty low and maybe too low to actually be causing this effect that we're seeing across these wetland cells. All right, now, although this field experiment was inconclusive in terms of the effect of temperature on parasite transmission to snail hosts, it did provide us with genetically similar snails from the same cohort that were either uninfected or infected. And so this control allowed us to do another temperature performance experiment comparing the effects of host infection status on host growth, metabolism, and mortality. And for this experiment, we housed snails individually across just 3 temperatures. And we did this, and we didn't include these other more extreme temperatures in the experiment because remember, we only had 33 infected snails that we got from this field experiment. So we fed these snails a gelatinous mixture of chicken feed and fish flakes. And this food does not degrade or change weight in water. And so that allowed us to weigh food before and then a couple of days after to determine how much the snails ate. And we did this for 3 weeks. So we found that infected snails ate significantly more food, controlling for body size. And this increased with temperature for the infected snails, but not the uninfected snails. So up top, I'm showing you how much food remained at the end of the experiment for infected versus uninfected. And you see that the infected snails have eaten much more of their food than the uninfected. Additionally, we ended the experiment because we had such extremely high mortality of the infected snails. We found that after 8 weeks, infected snails had significantly higher mortality at all temperatures, but that mortality increased for the 2 highest temperatures with all of the snails dying. So it's pretty conclusive. So to wrap all this up of what we have learned about the system, how does temperature influence host parasite dynamics in our system? Well, for the host, there was a steep decline in survival and growth above 20 degrees Celsius. And snails also matured earlier and smaller at higher temperatures and had lower total fecundity. This suggests that under warmer temperatures, snails may have more time to reproduce before getting infected and castrated, but may not actually produce more offspring due to their lower fecundity.
4 minutes.
Thank you. Almost done. So in terms of the free-living egg Miracidia stage of our parasites, infection prevalence increased farther from the treatment plant, though that was not correlated with temperature at the microhabitat stage that we were measuring temperature at. We did find that infection prevalence increased with bird activity and decreased with ammonia, which are things we'll definitely follow up on. And for the interaction between hosts and parasites, infected snails ate more and the difference increased with temperature. And infected snails also had higher mortality, and this also increased with temperature. So under warmer conditions, infected snails may have larger impacts on their ecosystem in terms of foraging. However, this might be offset by their shorter lifespan. This spring, I'm hoping to kind of increase my field studies so that we can disentangle the factors influencing infection prevalence, and also so that we can get larger numbers of these infected snails to investigate this interaction more fully and do some follow-up experiments. And so I'd like to thank all my advisors, collaborators, and funding. And I will take questions.
Great. Thanks so much, Emlyn. You already have a question in the chat, so I'll relay that. And as people think of questions, feel free to send them in the Zoom chat window or through Slack. So your first question comes from Collin. Awesome talk. Is trematode dispersal influenced by water flow? Do you expect that as you move downstream in these wetlands, trematode densities will naturally increase?
So that has been something that has been suggested when I've talked to people about the system. I don't have a good sense for these specific trematode species of how long they live. That's some follow-up work that we need to do. And that should tell us how far they can actually disperse. These are pretty big ponds, and the water it is flowing, but very, very slowly. So that is a possibility, but it's not kind of high up there on, I guess, where I'm thinking of what's happening, but it's definitely something we need to follow up on.
Great, we have another question for you from Joey. Awesome talk, Emlyn. What explains the lower fecundity in the warmer snails that developed faster? Did they also die earlier, or did they get castrated earlier?
So we've taken in, so for the first experiment, there was no parasites, so nobody got castrated. Lucky for them. What I think is happening is there were the same number of egg masses between T3 and T4, those two temperatures that you're talking about, were the same, but T3, which is the cooler temperature, and the snails that matured at a larger size, their egg masses had a lot more eggs per clutch. So even though they during the experiment didn't get to reproduce as long because they matured slightly later, they made up for it by a lot in the size of their egg clutches.
Great, so I think that we should move on to the next talk. Another really excellent talk, thank you, Emlyn. Our next talk today is from Christopher Kozma, and the title of his talk is The Structure and Stability of Moth Pollination Networks Along an Elevational Gradient, Insights into the Effects of Climate Change on Plant Pollinator Communities. Go for it.
Hi, yeah, thanks so much for the introduction. I'm Chris Kozma. I'm a PhD student in Nicole Rafferty's lab at UC Riverside. And today I'm going to talk a little bit about moth pollination and climate change. So this is an ongoing project, but I am excited to share some preliminary results with you today. So first, some quick background. Plant-pollinator interactions are an important ecosystem service. They contribute to the sexual reproduction of about 85% of all angiosperm species, as well as increased fruit production in about 35% of our food crops. Along with other anthropogenic drivers, global climate change is disrupting plant-pollinator interactions. And this can occur through a variety of mechanisms, among them through physiological effects on both plants and pollinators, so things like reduced nectar in plants and altered pollinator behavior. And this can also occur through independent shifts in space and temporal activity in plants and pollinators. And as a result, interactions become an interaction mismatch, and this may lead to potential population declines in both plants and pollinators, since both rely on the resources exchanged through interactions in order for successful reproduction. It's important to recognize that species and pairwise interactions are not isolated in their response to climate change. Instead, they're connected in a larger network of interactions between other species in the community. And ecological networks have provided a community-level approach to the study of plant-pollinator interactions. And such studies have identified structural features such as nestedness and modularity in plant-pollinator communities that help determine the community's stability and response to environmental disturbances, as well as the loss of species and interactions. Another approach to study plant-pollinator interactions and climate change is by using natural experiments provided by environmental gradients. For example, elevational gradients provide a space for time substitution where network structure can be analyzed in relation to the climatic variation along these gradients in order to make predictions about how climate change may shift network structure and stability. But the vast majority of these studies have focused on diurnal pollinators and specifically bees, which leaves our understanding of nocturnal pollinators and their community interactions behind. So moths have long been known to be important nocturnal pollinators, and in many cases they can perform different functions than diurnal pollinators such as bees. So because moths' pollination activity is not tied to nest sites like bees, they can act as long-distance pollen dispersers. And this is important for maintaining gene flow between plant populations. Furthermore, moth network patterns may be constrained by different factors in diurnal networks, for example, the distribution of larval host plants. And finally, moths face many of the same threats that diurnal pollinators do, such as habitat fragmentation and climate change, as well as unique threats like increased light pollution. And a combination of these factors is leading to the decline in the diversity and abundance of moths globally. Now, historically, most moth pollination research is focused on highly specialized pairwise interactions, such as the yucca-yucca moth interaction. However, a growing number of studies at the community level, so using pollination networks, are beginning to reveal that moth pollination performs a much larger community role. So moths actually pollinate entire communities of plants, and in many cases, they provide redundant interactions to some of the same plants that are pollinated during the daytime by bees and other diurnal pollinators. And by providing these redundant interactions to these plants, moths may increase the robustness of these communities to climate change and other perturbations. Now, most of these studies are coming out of the UK, where they're finding between 24 and 46% of moths are transporting pollen in these networks. And even more strikingly, a series of studies from Portugal has shown between 70 and 76% of moths are transporting pollen in these communities, and in some cases from the majority of the flowering species that are present at these sites. But because these network studies have been localized to Europe, our view of moth pollination networks may be slightly biased. For example, there's a notable lack of data on community level moth pollination from the United States. So that brings me to my research. I study moth pollination in Southern California, specifically along an elevational gradient that begins at Boyd Deep Canyon Desert Research Center, which is a UC natural reserve in the Colorado Desert. The elevational gradient extends up into the Santa Rosa Mountains and spans about 2,400 meters in elevation. It also encompasses multiple distinct habitat types, and as well as containing over 700 different plant species. Besides being an absolutely beautiful area to conduct research, this location is ideal to study the effects of climate change and the space for time substitution. And that's because we have these really nice temperature and precipitation trends going on along this gradient. So as elevation increases on the x-axis, we see precipitation in the blue bars increases quite drastically and temperature decreases. Another reason why it's an ideal location to study nocturnal pollination is because in general, there's a greater prevalence of nocturnal pollination interactions in xeric environments. And a review paper by Borges et al. 2016 proposed the arid or water stress hypothesis of nocturnal flowering and thereby pollination, which follows that nocturnal flowering is an adaptive strategy for plants to avoid water loss through evapotranspiration, and nocturnal flight is an adaptation for insects to avoid high temperatures and low humidity during the day. And so in the dry habitats across the Southwest, there's a high diversity of night blooming plants, as well as a high diversity of pollinating moths. So that brings me to the questions, and today I'll be focusing on one question since this is preliminary data, and that question is, what is the community level contribution of moth pollination and the distinct habitat types along this elevational gradient? So following that arid or water stress hypothesis, I propose that plants in drier environments may rely more on nocturnal pollinators and therefore predict that a higher percentage of the moths in the drier low elevations will be carrying pollen. So I'll take you through my methods now. I sample moth communities using light traps. I then identify the moths to species level. I remove the pollen loads from the proboscis of these moths, and I perform DNA metabarcoding on these pollen loads in order to detect interactions with plants. Finally, that allows me to construct and model networks. So I chose 5 transects out of a system of 21 transects that have been used to study the effects of climate change on plant community composition. And these 5 transects encompass both the elevational range as well as the distinct habitat types along this gradient. So they begin at the lowest elevation in the low elevation desert here at the bottom and continue up in elevation to the conifer forests in the Santa Rosa Mountains. And along each of these 400-meter transects, I've established 3 study locations that are separated by at least 100 meters. And each night of data collection, I randomly deploy 3 light traps to each of these locations, and I run these traps approximately from sunset to sunrise each night. So here's a result, a summary of the 2020 field seasons. Going through this figure, we have the date on the x-axis and the elevation on the y-axis. And so I began at the lowest elevation in February 2020 and followed flowering Phenology as it progressed up in elevation, ending at the highest transects in July. And at several of these transects, notably the 840-meter transect, I was able to sample much more thoroughly through time as well. And so here's what my homemade moth traps look like, as well as what a typical night of collection may look like in these traps. So you can see that the abundance of moths is quite high at these locations. And so the next step is identification. And by running side traps simultaneously each night, I was able to construct reference collections of moths from each location and time point. And using these, I've been identifying moths to species level with the help of a local expert. And that is still ongoing, but I do have some preliminary results. So here's a graph displaying the moth species richness by elevation with elevation on the x-axis. We can see that there's a relatively high diversity of moths at these transects. So peaking at about 40 species at this 2290-meter transect. And here's that same data displayed along the elevational gradient. So I'm still processing samples from this low elevation, but we can see again, these more intermediate elevations have a relatively higher diversity of moths. And here's that same data broken down by moth family. So the number of species per moth family at each elevation, with elevation again on the x-axis. And you can see across the board here, this family, Noctuidae, in the orange dominates, especially at these highest elevations. And that's to be expected because Noctuidae is the most species-rich family in California. And interestingly, these top 4 families, Noctuidae, Erebidae, Geometridae, and Sphingidae, are the moth families that are the best known moth pollinators. So particularly Sphingidae, which are these large, robust hawk moths, are actually used as a study system in pollination research. And so this figure is a heat map showing the number of shared species between elevational sites. And so the raw species number is recorded in the boxes, with warmer colors corresponding with relatively more species being shared. And so you can see a general pattern that these higher elevation sites, so the 2450 and 2290 meter transects, are sharing relatively more species than these lower transects. And that's partly because these transects are closer together to one another than these lower ones are. And so that same data put as a proportion of the total species shared between the transects, we can see that there's a relatively high species turnover going on along this gradient, with only about a maximum of 20% of species being shared between any two elevational sites. And as we would expect, the species that are shared between elevations tend to be habitat generalists. So an example of that is the hawk moth Hyles lineata, which is a relatively large, robust flyer. So it has a high dispersal ability, and it's been shown to forage across a variety of different habitat types. And it's these types of moths that may be playing important roles in connecting plant populations for maintaining gene flow. And so next I'm going to go through the preliminary results for pollen transport. And this is what the pollen looks like under a dissection scope on the proboscis of these moths. So this is preliminary data. I'm still performing the metabarcoding, so I don't have the actual network data with the plant species. However, I'll take you through the results that I do have. So this graph displays the percent of moths carrying pollen at each elevation. And we can see a relatively high percentage of moths transporting pollen, peaking at about 70% at this lowest elevation. And again, here's that same data along the elevational gradient. So we have about 66.7% of moths at this lowest elevation transect, and that percentage of moths transporting pollen steadily declines going up in elevation. And again, this is pollen visible under a dissection scope. So once the metabarcoding results come out, I have a feeling that there's a lot of pollen that I can't see under the dissection scope that's going to be identified. And so that summarizes my preliminary results. And circling back to that first question that I asked, what is the community level contribution of moth pollination in the distinct habitat types along this gradient? The results so far seem to suggest that my prediction that a higher percentage of moths in the drier low elevations will be carrying pollen is supported. Again, this is preliminary data, but if this pattern persists, a possible climate change prediction may be that as predictions become hotter and drier, selection pressures may shift to favor nocturnal pollination interactions. So what remains to be done on this project, I need to complete the identification of these moth specimens, as well as continue the pollen DNA metabarcoding, which will ultimately allow me to construct and model these networks in relation to the climatic variation along this gradient. And so that by constructing these networks, I'll be able to address the second question: how does the structure and stability of moth networks shift along the elevational gradient in relation to those climatic factors? And it's by answering this question that I'll be able to form predictions about how climate change may shift the structure and stability of these networks. And so following this study, I will repeat the same sampling procedure during this field season in order to quantify the interannual variation, which has been shown to be an important dynamic in plant pollinator communities. And I'm also working on adding phylogenetic information in order to examine the evolutionary and co-evolutionary mechanisms that may be structuring these networks. So with that, I'd like to thank everyone who's helped me so much on this project, in particular my advisor, Nicole Rafferty, and the rest of the Rafferty lab, as well as my funding sources. And I can take any questions.
Great, thanks, Christopher. So for people who have questions, feel free to send them to me in the Zoom chat window or use the Slack channel. We have about 4 minutes for questions. Okay, we have a question for you, Chris, from Judy Bronstein. Great data. Do you know if any of the moths do carry? Do you know if any of the moths do carry pollen but do not transfer it? You're equating pollen carrying with pollen delivery, which is probably generally true, but there might be some important exceptions.
Yeah, that's a really good question. I think it's one of the setbacks to these types of studies. Of course, as you mentioned, pollen transport doesn't necessarily mean successful pollination. And so we know from a variety of study systems, such as the hawk moths, that some of these moths are functioning as important pollinators and resulting in plant seed set. As far as community level patterns, that's hard to say. And so I'm actually coupling this study with the pollinator exclusion experiment, in which I'll basically be quantifying the relative contribution of nocturnal and diurnal pollination interactions to some of the plants along the gradient. So that's one way that we can start to tease apart the actual functional consequences of these interactions. But yeah, it's a good question and one that I'll be thinking about more in the future.
Great, we have another question, this one coming from the Slack channel from Bob Week. Awesome talk. How much does the community composition change across the elevation gradients considered? And how does this interact with your time for space study of the effects of climate change on the stability and structure of networks?
Yeah, another really interesting question. I would have to ask, are you talking about the plant community or the moth community? So if you're talking about the plant community composition, I'm by no means an expert on that research. So a couple of really high profile papers have come out of this same gradient, including Kelly and Gould in 2008. And I believe that many of the plant species are shared between elevations, and I can say that with certainty because I work along these gradients. So some species, some of the dominant like shrub species, perennials, such as creosote, have a relatively wide elevational range. So the plant community it depends on the plants that you're looking at. So some of the desert annuals I think are a little bit more variable, but as far as how this interacts with my study system, I think that's a really important question too, because particularly with Lepidoptera, since they have these really tight host plant relationships in their larval stages, I'm expecting to see a pretty large effect of the plant community on these moths. And so as pollinators, of course, like I mentioned, they may have a higher dispersal ability even compared to some other pollinating taxa. So I'm not sure how much that would translate into the adult stages of these moths, but yeah, I don't know if that answered your question really, but I think, yeah, if you check out that paper by Kelly and Goulden, it's a really cool paper.
Awesome, thanks, Chris. So you can look at the Slack channel, and Bob was interested in both communities, so you can maybe reply there, and I'll send you one other question from the chat.
Okay, so thanks so much, Chris. That was such a great talk. We are nearing the end of our session. We have our final talk of the day. I just want to really quickly thank all of the excellent speakers we've heard from today. I just think it's been such a exciting day in the middle of a really hard time, and it's been really fun to see all of these really exciting ideas and hear from all of these scientists, many young scientists here today. All right, so our final talk of the day is from Anna Parker, and she'll be giving a talk entitled "Very Hungry Caterpillars: A Combination of Parasitization and Heat Stress Increases Consumption of Host Plants by Manduca sexta." Go for it.
Thank you so much. And thank you to everybody for sticking it out through this session all the way to the very end to see my talk. So yes, as you mentioned, I'm going to be talking about some very hungry caterpillars and their effects on my tritrophic system. So studying tritrophic systems or systems where there are interactions between an herbivore, its food source, and its natural enemy can be very illustrative when we want to understand how broader communities might respond to changing biotic or abiotic conditions. So if we look at, say, this bird, the fuzzy caterpillar it's eating, and the maple tree that the caterpillar is consuming, and we perturb the system somehow, we might be able to predict how the broader forest community would respond to the same perturbation. And that's because we're capturing 3 different trophic levels, their interactions, in this microcosm of a system that we're investigating. And communities are built out of chains of these multitrophic interactions. So one such perturbation affecting our natural world is global warming, and specifically increasing daily mean and high temperatures. Here you can see data from the 2017 US Climate Report showing an increase in the number of record daily high temperatures recorded, especially in the past 20 years. And in the King Solver Lab, we often study the effects of increasing temperature using the model organism Manduca sexta. In recent years, my lab mate Elizabeth Moore, who you heard from earlier this session, has added to our research the most prevalent natural enemy of Manduca sexta in the southeastern United States, Cotesia congregata. This parasitoid wasp lays its eggs in Manduca sexta, and it also deposits venom and an endogenous virus that disrupts the host's immune system and developmental pathways. This allows the wasp larva to develop uninterrupted inside the host before emerging through the cuticle, spinning a cocoon, and eclosing as adult wasps. I have expanded this system to include a common host plant of Manduca sexta, tobacco. And in the experiment I'm going to share with you today, I investigated how 2 perturbations to this system affected the life history traits of its members. And the first perturbation I've already mentioned, it's temperature. And the second perturbation is the addition of a novel host plant, Proboscidea louisianica, or devil's claw. And this plant has been shown to be an alternate host of Manduca sexta in the southwestern United States. But it has recently invaded the southeast US, presenting an opportunity for Manduca to utilize it here as well. And previous work in our lab by Sarah Diamond has shown that devil's claw is a lower quality host than tobacco for Manduca sexta. It has negative effects on the caterpillars, including slowing down their development and minimizing their immune response. However, its potential to affect Cotesia congregata, mediated through this caterpillar host, is unknown. So in this experiment, I set out to answer the question, how will the life history traits of each trophic level be affected by increased temperature in the form of a heat shock and the introduction of a novel host plant? Now I'm going to step through my experimental design before coming back to the life history traits I'm actually measuring. So I reared caterpillars on tobacco. On the day they molted to 3rd instar, I either parasitized them or left them unparasitized. And that same day, I then either placed them into a heat shock of 40 degrees Celsius for 12 hours, or I left them at their rearing temperature of 25 degrees Celsius. So this created 4 different treatment groups, and we expect that the developmental outcomes for these caterpillars are going to differ based on treatment group. So for my non-parasitized, non-heat shocked caterpillars, we expect them to undergo normal development, reaching a state called wandering, which is right before they pupate and become an adult moth. For the non-parasitized heat shocked group, we still expect them to reach wandering, but perhaps with an effect on mass or development time caused by that 12-hour heat shock. For my parasitized non-heat shocked group, we expect the wasps to undergo normal wasp development, with larvae developing, emerging, and eclosing from the caterpillar hosts. However, that injected virus will still shut down the caterpillar development, so caterpillars in this group will inevitably stop feeding and die after wasp emergence. Now for the parasitized heat shocked group. Previous work by Elizabeth Moore again has identified a thermal mismatch between the caterpillar hosts and the wasp larva. If you heat shock directly after parasitization, as I have done here, it can result in total wasp mortality. However, the virus is still active, meaning that the caterpillars don't undergo normal development. Instead, we see a host without wasp emergence, or a WOWI, that continues to feed and grow bigger and bigger until they rot. This outcome is what we expect to see out of this parasitized heat-shocked group. So I then mirrored these treatment groups on developmental outcomes on caterpillar hosts reared on devil's claw, the lower quality host plant. So we'll see if the lower quality host affects the developmental outcomes we see. Now back to what I'm actually measuring. For each trophic level, I will be measuring life history traits that are relevant to their survival and fitness. For the Manduca sexta, I will see how parasitization, heat shock status, and host plant all interact to affect growth and developmental outcome. For the host plants, the metric I'm measuring is leaf consumption. And I'm interested to see how treatment group, so parasitization and heat shock status, mediated through the caterpillars, influences how much these caterpillars eat. And I'm especially interested to see if consumption differs between the two host plant types. Finally, for the wasps, the metric I'm measuring is survival to eclosion. So I'm mainly seeing whether host plant mediated through the caterpillar affects survival, since I'm only expecting to see wasp emergence out of one of my treatment groups. Now I'm going to dive into my results. My notation will be consistent throughout my graphs, so I'm going to run through that really quickly first. So color is going to denote the host plant the caterpillars consumed, with red denoting devil's claw and blue denoting tobacco. Symbol type is going to represent parasitization status, with triangles representing parasitized individuals and circles representing unparasitized individuals. And then finally, heat shock status is going to be reflected by the closed or open nature of those symbols. So a filled in symbol is going to mean the individual was heat shocked, and an open symbol is going to mean the individual was not. So first, I'm showing you my caterpillar data for my developmental metrics. So on my y-axis, I have the maximum mass achieved by the caterpillar, and on the y-axis, and on the x-axis, I have the developmental time at that maximum mass. So right now, I'm showing you my unparasitized treatment groups, both those that were heat shocked with these closed circles and not heat shocked with the open circles. And as you can see, there's no difference in development time or mass with heat shock status. And so for this treatment group, this maximum mass is going to be at wandering. But there is a developmental time delay for the individuals reaching wandering who have been feeding on devil's claw, as you can see with these red points clustered to the right of the graph. But there is not a difference in maximum mass achieved at that point based on host plant. Now I'm going to add in my parasitized non-heat shocked treatment group. So for these guys, maximum mass achieved is right at parasitoid emergence. And again, we're not seeing an effect of host plant on mass achieved. There might be a slight delay, but it's not as large as in the wandering treatment groups. And finally, I'm going to add in my parasitized heat shocked group. Now, these are kind of all over the place. Let me start up here. These hosts up here are my WOWI hosts that I expected to see. Because of that stalled development from the wasp virus, they take longer, but they also reach larger mass. And these ones are the ones that keep eating and eating, and these are the ones I expected to see. However, I also have this group down here, which resembles the parasitized non-heat shocked caterpillars. And I did not expect to see these. Some of these had wasp emergence, indicating that the heat shock I used didn't kill all of the wasp larvae. And some of them just stayed small. They didn't have wasp emergence, but they just stayed small. They stopped feeding early on in their development, and they just never got big. And based on some very preliminary results by Elizabeth, we expect that these small WOWY hosts, when dissected, are actually going to contain partially developed wasp larvae, while these large WOWY hosts, when dissected, will have no discernible larvae. So we think that these larvae are partially developing, preventing this host from getting huge, but then not actually making it out through the cuticle to emergence. And again, we do see an effect of host plant on development time, both in my large WOWIes and my small WOWIes, but no real effect of host plant on mass. So now I'm going to move on to my my plant life history traits with leaf consumption. So this graph is going to be parallel to these previous ones, except now on my x-axis, I have leaf surface area consumed in square centimeters. So again, I'm starting with my 2 unparasitized treatment groups. Again, there's no difference in heat shock status. There's no difference in host plant. And these ones ate about 2,000 square centimeters. That's about the size of your laptop if you open it up flat. Now I'm going to add in my parasitized non-heat shocked group. These guys consumed far less and grew less. And you can see that there's a nice relationship forming here between leaf area consumed and maximum mass achieved. And this makes sense. The more you eat, the more you grow. Finally, I'm going to add in my parasitized heat shock group. And again, those are spread kind of along my whole distribution with a big cluster down here at the bottom and a big cluster up here at the top. And these clusters down here at the bottom with these parasitized non-heat shocked group, they ate about the size of a dinner plate worth of leaf. Whereas these guys up here that ate upwards of 5,000 square centimeters, they're eating the size of like a child desk, like a school desk, which is a lot for a little caterpillar. And again, I'm not seeing an effect of host plant on any of these treatment groups. So they're consuming equal amounts of tobacco and of devil's claw. So finally, I'm going to show you my wasp results, my wasp survival results. And now these graphs are going to have different axes. On the x-axis, I have the number of wasp larvae emerged per caterpillar host. And on the y-axis, I have the proportion of those emerged wasps that eclosed. So this is a conditional survival probability based on the number of wasps that emerged, how many of those, what proportion of those eclosed. And you can see that I don't have very many filled in triangles. I only had 6 individuals in my parasitized heat shocked group that had wasp emergence. And so even though it's a small sample size, I still have this trend where 4 out of my 6 individuals had very low numbers of wasps that emerged. So this is, again, the individuals that have undergone this heat shock. It seems like the heat shock has killed most of the wasp larvae and only a few are making it out. And I see no effect of host plant on my parasitized non-heat shocked group. The tobacco and devil's claw individuals seem to be fostering the same number of wasps that emerge and eclose. So that was a lot. So I'm going to summarize it all for you. So basically my treatment group, which is my combination of parasitization and heat shock status, was relevant to all 4 of the developmental metrics I measured, or the life history traits I measured. And host plant was only relevant for caterpillar development time. And this was a pretty interesting result. Since devil's claw is a lower quality host for Manduka, we expected to see cascading effects upwards to the wasps. These treatment groups produced these different developmental outcomes, and that was really what drove my results, which makes sense. We were trying to make different developmental outcomes with these treatment groups. So for my conclusions, this thermal mismatch between herbivores and their natural enemies can have consequences for all trophic levels of the system, including the host plants. And as daily maximum temperatures increase, we expect to see these effects occurring more frequently in our terrestrial ecosystems. For my future directions, I'm going to be conducting a field experiment this summer to assess the effects of the differential consumption that I saw on the host plants. So I'm really interested in nailing down effects on host plant fitness. So I'm going to be measuring above and below ground biomass and flower and seed set post-consumption to investigate further how these different developmental outcomes influence the host plants. And with that, I'd like to thank my lab members and some undergraduate assistants, especially Amy and Tyler, who helped me collect some of this consumption data. I'd like to thank my committee and my funding sources. And with that, I will take any questions.
Great, thanks so much, Anna. So again, for people who have questions for Anna, you can send them my way in the Slack channel or through Zoom. Okay, we have a question from Judy Bronstein. Why don't the wowies emerge as adults? And in parentheses, great talk.
Thanks. So I was a little bit untruthful in my statement. Sometimes the wowies do undergo host rescue. So in a couple circumstances, we've seen the individuals kind of present as a wowie, eat more. Sometimes they molt into an additional instar. And then a couple weeks into their last instar, they will actually wander. Most of those don't pupate successfully. They die as pupal intermediates. But there's actually another study by another one of my lab members, Catherine Malinsky, that's investigating the process of that host rescue. We think that the majority of them don't wander because the virus is still active and it shuts down the wandering in a normal parasitized individual because the wasp larva can't chew through the pupal casing. So they need to emerge before the wasp or the caterpillar has pupated. So yeah, we see rescue in about 15 to 20% of our hosts. I just didn't happen to see any rescue in my experiment.
Any other questions for Anna? Oh, we do. Have you altered the duration of heat shock to see if longer heat shock might alter the outcome? This is from Emlyn.
Yes. So I chose a 12-hour heat shock because I was feeding them leaf material and the leaves kind of desiccated in our growth chambers when I performed the heat shock for longer periods of time. But we've actually produced wowies under 4 different heat shock regimes. 6 hours at 40 degrees Celsius, 12, 24, and then also at a fluctuating heat shock where they soak for 2 hours at 42 degrees for 3 consecutive days post-parasitization. So I did a follow-up, because I got so many small wowies, I did a follow-up short little thing to this study where I shocked them for 24 and for 12. And I did get a higher proportion of large wowies in my 24-hour heat shock. But 12 hours is a little bit more ecologically relevant, and it allowed me to better measure consumption with my live leaves I was using. Good question.
Great, thanks, Anna. I think that wraps up the session.
Yeah, just another round of applause for all of our speakers and for everyone for spending the whole day with us. Thank you, everyone, so much. Have a good evening or morning or wherever you are.